Hi there, preparing for D312. OA exam can feel overwhelming, but it doesn't have to be. Stick with us, and we're going to help you with it. We built a free question bank for the D312 OA exam. Preparing for an OA is no joke, so our guides and practice questions are completely free. In this video, we will cover questions from skeletal system, muscular physiology, neural integration, endocrine control, special senses. So, let's get started.
1. A researcher examining a prepared cross-section of compact bone tissue observes concentric rings of calcified matrix arranged around a central channel containing blood vessels and nerves. Which structural unit of bone is this researcher most likely identifying in this prepared-to-tissue sample? A. A trabecula within spongy bone. B. A lacuna housing an osteocyte. C. An osteon within compact bone. D. A canaliculus connecting bone cells. Do you know the answer? Correct answer, C. An osteon within compact bone. Explanation. Option C is correct, because the osteon is the structural unit of compact bone, defined by concentric lamellae of calcified matrix surrounding a central Haversian canal that carries blood vessels, nerves, and lymphatic vessels. Option A is incorrect because trabeculae are branching plates found in spongy bone that form a lattice-like structure. They do not contain osteons or concentric rings of matrix around central canals. Option B is incorrect because a lacuna is a small space within bone matrix that houses a single osteocyte. It is a tiny cavity, not a large structural unit with concentric rings. Option D is incorrect because canaliculi are tiny channels extending from lacunae that allow osteocytes to communicate with each other. They are microscopic passages, not the structural unit described with concentric lamellae. Alright, let's get into the building blocks of bone. This question is about compact bone tissue and its structural unit, the osteon. Picture a tree trunk cut crosswise. You'd see rings, right? An osteon looks like that. It's a set of concentric rings made of calcified matrix called lamellae wrapped around a central canal, the Haversian canal, which carries blood vessels, nerves, and lymphatic vessels. That's the defining feature, concentric lamella around a central canal. Now, why not the other options? A trabecula is a plate of spongy bone, no concentric rings, no central canal. A lacuna is just a tiny pocket where a single osteocyte sits. A canaliculus is a microscopic channel that lets osteocytes talk to each other. It's a passage, not a structural unit. So when the question says concentric rings around a central channel with blood vessels and nerves, that's an osteon every time. Here's a WGU D312 OA exam tip for you. When you see concentric rings or central canal in a question, your brain should snap to osteon immediately. Quick recall hook, think osteon, O-rings. The O in osteon looks like the rings it's made of. 2. A bone specimen from a healing fracture shows active new bone tissue being deposited along the inner surface of the periosteum during the repair process. Which type of bone cell is primarily responsible for forming this new bone matrix at the repair site? A. Osteocytes trapped in lacunae. B. Osteogenic cells deep in marrow. C. Chondrocytes within the periosteum. D. Osteoblasts within the periosteum. Do you know the answer? Correct answer, D. Osteoblasts within the periosteum. Explanation. Option D is correct because osteoblasts are the bone-forming cells located in the periosteum and endosteum. They actively produce and deposit new bone matrix, which is their primary function during growth and fracture repair. Option A is incorrect because osteocytes are mature bone cells already trapped within lacunae in the existing bone matrix. Their primary role is maintaining mineral concentration, not actively forming new bone tissue. Option B is incorrect because osteogenic cells are stem cells that can develop into osteoblasts but are precursors to bone-forming cells. They themselves do not directly deposit the new bone matrix during repair. Option C is incorrect because chondrocytes are cartilage cells, not bone cells. They are embedded in cartilage matrix and are not typically found in the periosteum or involved in direct bone matrix formation. This question zeroes in on which bone cell actually builds new bone. The answer is the osteoblast. During fracture repair, osteoblasts sitting in the inner layer of the periosteum go to work laying down fresh bone matrix. That inner layer is called the osteogenic layer. It's where bone formation happens. Now, osteocytes are mature bone cells already locked inside lacunae. They maintain the matrix that's already there, but they're done building. Osteogenic cells are the stem cells that become osteoblasts. They're the precursors, not the builders themselves. And chondrocytes, those are cartilage cells. They live in cartilage matrix, not bone. The question specifically says new bone tissue being deposited along the inner surface of the periosteum, and that's osteoblast territory. Here's a WGU D312 OA exam tip for you. If a question mentions bone formation or bone deposition, look for osteoblast. The word blast means building. Osteoblast builds bone. Quick hook, blast =, build. 3. In an adult patient, a physician orders imaging of the femur and notes that the hollow central portion of the shaft is filled with a fatty substance rather than blood-forming tissue. Which structure and its contents are being described in this clinical imaging finding? A. Medullary cavity with yellow marrow. B. Epiphysis filled with red marrow. C. Periosteum containing osteoblasts. D. Spongy bone housing trabeculae. What do you think is the answer? Correct answer, A. Medullary cavity with yellow marrow. Explanation. Option A is correct because the medullary cavity is the hollow space inside the diaphysis of a long bone. In adults, it is filled with yellow marrow, which consists primarily of stored adipose fat tissue. Option B is incorrect because the epiphysis is the widened end of a long bone filled with spongy bone and red marrow, not the hollow shaft region. Red marrow produces blood cells rather than fat. Option C is incorrect because the periosteum is the fibrous membrane covering the outer surface of the bone. It contains osteoblasts and osteoclasts, but is not a hollow cavity filled with any marrow. Option D is incorrect because spongy bone is a lattice of bony plates called trabeculae, found mainly in the epiphyses. It is not a hollow cavity, and its spaces contain red marrow, not yellow marrow. Here we're looking at the inside of a long bone shaft, the diaphysis. In an adult, the hollow centre of that shaft is called the medullary cavity, and it's packed with yellow marrow. Yellow marrow is basically stored fat. That's the fatty substance the imaging shows. Now, the epiphysis is the wide end of the bone, and it contains spongy bone with red marrow. That's where blood cells get made. The periosteum is the membrane on the outside of the bone. It's not hollow, and it doesn't store marrow. Spongy bone with trabeculae sits inside the epiphyses, not in the hollow shaft. So when you read hollow central portion of the shaft and fatty substance, that's medullary cavity plus yellow marrow, period. Here's a WGU D312 OA exam tip for you. Yellow marrow equals fat, red marrow equals blood cell production. If a question says fatty or adipose inside a bone shaft, you're looking at yellow marrow in the medullary cavity. Quick hook, yellow =, yummy fat stored away. 4. A teenager experiences a fracture through the region of hyaline cartilage located between the diaphysis and epiphysis of the tibia. The physician expresses concern that this injury could permanently affect future bone lengthening. Which structure has most likely been damaged by this specific fracture? A. The periosteum covering the shaft. B. The epiphyseal plate in the metaphysis. C. The endosteum lining the cavity. D. The articular cartilage at the joint. What do you think is the answer? Correct answer. B. The epiphyseal plate in the metaphysis. Explanation. Option B is correct because the epiphyseal plate is the growth plate made of hyaline cartilage located in the metaphysis between the diaphysis and epiphysis. Damage to this plate can disrupt longitudinal bone growth permanently. Option A is incorrect because the periosteum is the outer fibrous covering of the bone shaft. While important for bone repair and appositional growth, it is not the hyaline cartilage structure responsible for bone lengthening. Option C is incorrect because the endosteum is a thin membrane lining the medullary cavity inside the bone. It is not made of hyaline cartilage and is not located between the diaphysis and the epiphysis. Option D is incorrect because articular cartilage covers the joint surfaces at the ends of bones to reduce friction. It is not the growth plate located between the diaphysis and epiphysis that drives lengthening. This one is about a teenager who fractured the growth plate. The growth plate, formerly the epiphyseal plate, is a sheet of hyaline cartilage sitting in the metaphysis, which is the region between the shaft and the end of the bone. This cartilage is where bones grow longer. Cells in the plate keep dividing and getting replaced by bone until you stop growing, at which point it hardens into the epiphyseal line. If a fracture runs through this plate, it can permanently stop the bone from lengthening on that side. Now, the periosteum covers the outside of the shaft. It helps with bone width, not length. The endosteum lines the inside of the medullary cavity, nowhere near the growth plate. Articular cartilage sits on the joint surface for friction reduction. It has nothing to do with lengthening. Here's a WGU D312 OA exam tip for you. Anytime a question mentions a young patient, hyaline cartilage between the shaft and the end of a bone, and concern about growth, that's the epiphyseal plate. Quick hook. Plate equal sign penny. Place where bones get longer. 5. During a surgical procedure on the tibia, a surgeon carefully cuts through a tough fibrous membrane on the outer bone surface and notes that its inner layer contains cells capable of forming new bone tissue. Which membrane is the surgeon cutting through during this particular procedure? A. The endosteum of the medullary cavity. B. The dura mater surrounding the structure. C. The periosteum with its osteogenic layer. D. The perichondrium covering cartilage tissue. Do you know the answer? Correct answer, C. The periosteum with its osteogenic layer. Explanation. Option C is correct because the periosteum is the fibrous membrane covering the outer surface of bones. Its inner osteogenic layer contains osteoblasts and osteoclasts, which are the cells responsible for bone formation and remodeling. Option A is incorrect because the endosteum is a thin membrane that lines the internal surfaces of bone, including the medullary cavity. It is not located on the outer surface where the surgeon is making incisions. Option B is incorrect because the dura mater is the outermost meningeal layer that protects the brain and spinal cord inside the skull and vertebral canal. It does not cover long bones or contain osteoblasts. Option D is incorrect because the perichondrium is the membrane that covers cartilage, not bone. While structurally similar to the periosteum, it is found on cartilaginous structures rather than on bony surfaces like the tibia. A surgeon cuts through a tough fibrous membrane on the outside of a bone and finds that its inner layer has bone-forming cells. That's the periosteum. The periosteum wraps around the external surface of every bone. Its outer layer is dense, irregular connective tissue. That's the tough part the surgeon feels. Its inner layer is the osteogenic layer, packed with osteoblasts and osteoclasts that handle bone growth and remodeling. The endosteum is on the inside of the bone, lining the medullary cavity. A surgeon cutting from the outside wouldn't hit it first. The dura mater protects the brain and spinal cord, not long bones. The perichondrium covers cartilage, not bone. Here's a WGU D312 OA exam tip for you. Outer membrane on bone with an osteogenic layer. That's always the periosteum. If you see fibrous covering plus bone-forming cells, lock in periosteum. Quick hook, peri means around, osteum means bone, periosteum wraps around bone. 6. A clinician examines a patient and notes that the joint connecting two skull bones allows absolutely no visible movement and is held together by thin, fibrous connective tissue. Based on this clinical description, which classification best describes this type of joint articulation? A. A synarthrosis joined by fibrous tissue. B. An amphiarthrosis with cartilage. C. A diarthrosis with synovial cavity. D. A synovial joint allowing free movement. What do you think is the answer? Correct answer, A. A synarthrosis joined by fibrous tissue. Explanation. Option A is correct, because synarthroses are immovable joints held together by thin, fibrous connective tissue. Cranial sutures are the classic example, where skull bones are tightly joined with no movement permitted. Option B is incorrect because amphiarthroses are slightly movable joints connected by hyaline cartilage or fibrocartilage. These joints allow limited movement, unlike the immovable joints described. Option C is incorrect because diarthroses are freely movable synovial joints containing a fluid-filled synovial cavity. These joints provide a wide range of motion, not the lack of movement seen in the described joint. Option D is incorrect because synovial joints are the same as diarthroses and allow free movement with a synovial cavity. The joint in question is described as allowing no movement, which rules out synovial joints. This question tests whether you know how joints are classified by movement. The scenario describes a skull joint that allows zero movement, held together by thin, fibrous connective tissue. That's a synarthrosis. Synarthroses are immovable joints, and cranial sutures are the textbook example. Skull bones locked together by fibrous tissue with no play at all. An amphiarthrosis is slightly movable, connected by cartilage. Think of the vertebral discs or the pubic symphysis. A diarthrosis is a freely movable synovial joint, like your knee or shoulder. It has a fluid-filled cavity and allows a full range of motion. Synovial joint allowing free movement is just another way of saying diarthrosis, so that's wrong for the same reason. Here's a WGU D312 OA exam tip for you. When a question describes no movement and fibrous tissue, that's a synarthrosis. The prefix syn means together, and arthrosis means joint. Bones joined tightly together. Quick hook, syn =, stuck, no movement. 7. A physical therapist asks a patient to move their arm in a complete circular pattern at the shoulder, combining flexion, extension, abduction, and adduction into one continuous sweeping motion. Which specific type of body movement is the therapist asking the patient to demonstrate here? A. Pronation of the upper limb. B. Abduction away from the midline. C. Extension at the shoulder joint. D. Circumduction at the shoulder joint. What do you think is the answer? Correct answer, D. Circumduction at the shoulder joint. Explanation. Option D is correct because circumduction is a circular movement that combines flexion, extension, abduction, and adduction in a continuous cone-shaped motion. The shoulder and hip joints are common sites for this movement. Option A is incorrect because pronation is a specific rotational movement of the forearm that turns the palm to face downward or posteriorly and does not involve a full circular motion of the arm. Option B is incorrect because abduction is movement away from the midline of the body in a single plane and is only one component of the full circular motion being described by the therapist. Option C is incorrect because extension is the movement that increases the angle between bones at a joint. Like abduction, it is only one component of the complete circular pattern described in this scenario. The therapist asks the patient to swing their arm in a full circle at the shoulder. That motion, combining flexion, extension, abduction, and adduction into one continuous sweep is circumduction. It traces a cone shape in space. The shoulder and hip joints are the classic sites for it because they're ball and socket joints with multi-axial freedom. Pronation is just rotating the forearm so the palm faces down. No full circle involved. Abduction alone moves the arm away from the midline in one plane. Extension alone straightens or opens an angle at a joint. Neither of those covers the full circular sweep the question describes. Here's a WGU D312 OA exam tip for you. If a question describes a circular arm or leg motion combining multiple planes, that's circumduction. Quick hook. Circum equals sign circle. Circumduction equals sign circular motion at a joint. 8. A medical student studying joint classification observes that the elbow allows movement primarily in one plane through flexion and extension, while the hip permits movement in multiple planes and directions. What best explains the functional difference between these two specific synovial joints? A. The elbow is an amphiarthrosis joint. B. The elbow is a uniaxial hinge joint. C. The hip lacks a synovial cavity. D. The hip is classified as hinge type. Can you guess the answer? Correct answer, B. The elbow is a uniaxial hinge joint. Explanation. Option B is correct because the elbow is a hinge joint that moves in only one axis, allowing flexion and extension. The hip is a ball and socket joint that is multi-axial, permitting movement in many directions. Option A is incorrect because the elbow is a freely movable diarthrosis, not a slightly movable amphiarthrosis. Amphiarthroses include joints like vertebral discs that permit only limited movement between adjacent bones. Option C is incorrect because the hip absolutely contains a synovial cavity filled with lubricating fluid. It is classified as a diarthrosis and is one of the most freely movable joints in the entire body. Option D is incorrect because the hip is a ball and socket joint, not a hinge joint. Its rounded femoral head fits into the acetabulum, allowing multi-axial movement far beyond simple flexion and extension. This question compares two synovial joints, the elbow and the hip, and asks why one moves in just one plane while the other moves in multiple directions. The elbow is a hinge joint. Like a door hinge, it opens and closes in a single axis, flexion and extension. That makes it uniaxial. The hip is a ball and socket joint. The rounded head of the femur fits into the acetabulum, giving it multi-axial freedom. Flexion, extension, abduction, adduction, rotation, circumduction. Now the elbow is definitely not an amphiarthrosis. It's freely movable with a synovial cavity. The hip obviously has a synovial cavity too. Saying it lacks one is flat wrong, and calling the hip a hinge joint ignores its ball and socket anatomy. Here's a WGU D312 OA exam tip for you. Hinge equals sign uniaxial, ball and socket equals sign multiaxial. If a question compares limited versus wide range of motion, think hinge versus ball and socket. Quick hook. Hinge equals sign one direction, ball equals sign all directions. 9. A patient recovering from ankle surgery is asked to point their toes downward toward the floor as part of a rehabilitation exercise. The patient performs this movement successfully against light resistance from the therapist. Which specific ankle movement is this patient performing during the exercise? A. Dorsiflexion of the ankle joint. B. Eversion of the lateral foot. C. Plantar flexion of the ankle joint. D. Inversion of the medial foot. Do you know the answer? Correct answer, C. Plantar flexion of the ankle joint. Explanation. Option C is correct because plantar flexion is the movement of extending the ankle joint so the toes point downward, as when standing on tiptoes. This action is primarily performed by the gastrocnemius and soleus muscles. Option A is incorrect because dorsiflexion is the opposite movement, pulling the toes and foot upward toward the shin. This action is performed primarily by the tibialis anterior muscle, not when pointing toes downward. Option B is incorrect because eversion is the movement of shifting the lateral side of the foot, so the sole faces outward and away from the midline. It involves sideways tilting, not downward pointing. Option D is incorrect because inversion is the movement of lifting the medial side of the foot, so the sole turns inward toward the body midline. Like eversion, it involves sideways tilting rather than downward motion. The patient points their toes toward the floor. That's plantar flexion. Extending the ankle so the foot angles downward, like standing on tiptoes. The muscles doing the work are the gastrocnemius and soleus in the posterior lower leg. Dorsiflexion is the opposite, pulling the toes up toward the shin, done by the tibialis anterior. Eversion tilts the sole outward, away from the midline. Inversion tilts the sole inward toward the midline. Neither eversion nor inversion involves pointing toes down. They're side-to-side movements of the foot. Here's a WGU D312 OA exam tip for you. Toes down equals plantar flexion, toes up equals dorsiflexion. Think of it this way, you plant your foot on the floor during plantar flexion. Quick hook, plantar equal sign, plant your foot down. 10. During knee surgery, a surgeon identifies a band of tough, fibrous connective tissue that connects the femur directly to the tibia and helps prevent excessive movement at the joint. Which type of connective tissue structure is the surgeon most likely identifying in this procedure? A. A ligament connecting bone to bone. B. A tendon connecting muscle to bone. C. Cartilage covering the joint surface. D. The synovial membrane lining the joint. What do you think is the answer? Correct answer. A. A ligament connecting bone to bone. Explanation. Option A is correct because ligaments are bands of fibrous connective tissue that connect bone to bone at joints. Their primary function is to stabilize the joint and prevent excessive or abnormal movement. Option B is incorrect because tendons connect muscle to bone, not bone to bone. Tendons transmit the force of muscle contraction to the skeleton to produce movement rather than stabilizing the joint itself. Option C is incorrect because cartilage at the joint surface serves to reduce friction and absorb shock during movement. It does not form bands that connect one bone directly to another bone across a joint. Option D is incorrect because the synovial membrane is a thin tissue lining the inside of the joint capsule that produces synovial fluid. It does not form the tough, fibrous bands described in this scenario. The surgeon sees a tough, fibrous band connecting the femur to the tibia, stabilizing the knee and preventing excessive movement. That's a ligament. Ligaments are fibrous connective tissue that connect bone to bone at joints. Their whole job is joint stabilization. A tendon connects muscle to bone. It transmits force for movement, but it doesn't link two bones together. Cartilage covers joint surfaces to reduce friction and absorb shock. It's not a band connecting bones across a joint. The synovial membrane lines the inside of the joint capsule and makes synovial fluid. It's thin tissue, not a tough fibrous band. Here's a WGU D312 OA exam tip for you. Bone to bone equals ligament. Muscle to bone equals tendon. That's the distinction they'll test over and over. Quick hook. Ligament equals sign. Links bones. Tendon equals sign. Tugs on bone from muscle. 11. At the neuromuscular junction, a motor neuron action potential arrives at the axon terminal and triggers a specific sequence of events that ultimately leads to skeletal muscle fiber depolarization and contraction. Which substance is released first from the presynaptic terminal to initiate this entire process? A. Calcium from the sarcoplasmic reticulum. B. Sodium ions through the sarcolemma. C. Tropomyosin from the actin filament. D. Acetylcholine from the synaptic vesicles. Do you know the answer? Correct answer, D. acetylcholine from the synaptic vesicles. Explanation. Option D is correct because when a motor neuron action potential reaches the axon terminal, synaptic vesicles fuse with the presynaptic membrane and release acetylcholine into the synaptic cleft as the first chemical step. Option A is incorrect because calcium release from the sarcoplasmic reticulum occurs later during excitation-contraction coupling, after the muscle fiber membrane has already been depolarized by acetylcholine binding to its receptors. Option B is incorrect because sodium influx through the muscle fiber membrane occurs after acetylcholine binds to postsynaptic receptors. It is a response to the neurotransmitter, not the first event in the sequence. Option C is incorrect because tropomyosin shifting away from myosin binding sites on actin happens during the contraction phase after calcium has been released. It is not released from the presynaptic terminal at all. This question is about the very first chemical event at the neuromuscular junction. A motor neuron fires, the action potential races down to the axon terminal, and synaptic vesicles fuse with the presynaptic membrane. What spills out? Acetylcholine, AC. It crosses the synaptic cleft and binds receptors on the muscle fiber, which then lets sodium rush in and depolarize the membrane. Calcium release from the sarcoplasmic reticulum happens later, during excitation-contraction coupling inside the muscle fiber, not at the synapse. Sodium influx through the muscle membrane is a consequence of AC binding, not the first release event. Tropomyosin shifting on actin is even further downstream, during the actual contraction phase. The question asks what is released first from the presynaptic terminal, and that is always acetylcholine. Here's a WGU D312 OA exam tip for you. The neuromuscular junction sequence starts with HH release. If a question asks about the first event at the junction, it's AC leaving the presynaptic terminal. Quick hook, ACH equal sign. Action creates the handoff to the muscle. 12. After acetylcholine binds to receptors on the muscle fiber membrane and depolarization occurs, calcium is released inside the cell and binds to a specific regulatory protein on the thin filament, causing a conformational change. Which protein does calcium bind to directly during this step? A. Tropomyosin blocking the binding sites. B. Troponin on the actin filament. C. Myosin heads on the thick filaments. D. Acetylcholinesterase in the synaptic cleft. Can you guess the answer? Correct answer. B. Troponin on the actin filament. Explanation. Option B is correct because calcium release from the sarcoplasmic reticulum binds directly to troponin on the actin filament. This binding causes troponin to change shape, pulling tropomyosin away from the myosin binding sites. Option A is incorrect because tropomyosin does not directly bind calcium. It is pulled away from the myosin binding sites on actin only after calcium binds to troponin and causes that regulatory protein to change its shape. Option C is incorrect because myosin heads on the thick filaments do not bind calcium during the regulatory step. Myosin forms cross bridges with actin only after the binding sites are exposed by the troponin-tropomyosin shift. Option D is incorrect because acetylcholinesterase is the enzyme that breaks down acetylcholine in the synaptic cleft at the neuromuscular junction. It has no role in calcium binding or the regulation of cross-bridge formation. So AC has done its job, the muscle fibers depolarized, and now calcium floods out of the sarcoplasmic reticulum. Where does that calcium go? It binds directly to troponin, a regulatory protein sitting on the thin actin filament. When calcium locks onto troponin, troponin changes shape. That shape change drags tropomyosin off the myosin binding sites on actin, exposing them so myosin heads can grab on and start the power stroke. Tropomyosin itself doesn't bind calcium. It just gets pulled out of the way. Myosin heads form cross bridges with actin after the sites are exposed, not during the calcium binding step. And acetylcholinesterase is the enzyme that breaks down AC back at the synapse. It has zero involvement inside the muscle fiber. Here's a WGU D312 OA exam tip for you. Calcium binds troponin, troponin moves tropomyosin. That's the chain. If a question asks what calcium directly binds to, it's troponin. Quick hook. Calcium picks the troponin lock to open the door for myosin. 13. A histologist examining skeletal muscle tissue under high magnification identifies the contractile units within individual muscle fibers that are composed of repeating arrangements of thick myosin and thin actin filaments. Which term correctly identifies these specific contractile units found inside each muscle fiber? A. Fascicles wrapped in perimysium tissue. B. Endomysium surrounding each fiber. C. Myofibrils within the muscle fiber. D. Epimysium covering the whole muscle. What do you think is the answer? Correct answer, C. Myofibrils within the muscle fiber. Explanation. Option C is correct because myofibrils are the contractile units found within each skeletal muscle fiber. They're composed of organized arrangements of thick myosin filaments and thin actin filaments called myofilaments. Option A is incorrect because fascicles are bundles of multiple muscle fibers grouped together and wrapped in perimysium. They represent an organizational level above the individual fiber, not the contractile units inside it. Option B is incorrect because endomysium is a layer of loose connective tissue that wraps around individual muscle fibers from the outside. It provides structural support, but is not the contractile unit found inside the fiber. Option D is incorrect because epimysium is the outermost connective tissue layer that surrounds the entire muscle as a whole. It is the most external wrapping, not an internal contractile structure within individual muscle fibers. A histologist zooms in on a single skeletal muscle fiber and sees repeating units of thick myosin and thin actin filaments lined up inside it. Those are myofibrils, the contractile units within each muscle fiber. Each myofibril is made of organized myofilaments. Thick ones are myosin, thin ones are actin. That's where the actual sliding and contracting happens. Now, fascicles are bundles of multiple fibers wrapped in perimysium. They're a level above the individual fiber, not inside it. Endomysium is the connective tissue sheath around each individual fiber from the outside. Epimysium wraps the entire muscle as a whole. None of those three are inside the fiber. The contractile machinery inside the fiber is the myofibril. Here's a WGUD 312 OA exam tip for you. Remember the layers from smallest to largest. Myofilaments make up myofibrils. Myofibrils are inside fibers. Fibers form fascicles. Fascicles form the whole muscle. If a question asks about the contractile unit inside a fiber, that's the myofibril. Quick hook. Myofibril equals sign the fiber's internal machinery. 14. During the relaxation phase of skeletal muscle contraction, a specific mechanism must occur to stop cross-bridge cycling between actin and myosin filaments so that tropomyosin can return to its blocking position on the thin filament. Which event directly causes muscle relaxation to begin? A. Calcium pumped back into storage. B. Acetylcholine released at the junction. C. Sodium entering through that membrane. D. Troponin binding to additional calcium. What do you think is the answer? Correct answer, A. Calcium pumped back into storage. Explanation? Option A is correct because muscle relaxation begins when calcium is actively pumped back into the sarcoplasmic reticulum by calcium 8-pace pumps. Without calcium bound to troponin, tropomyosin slides back and blocks the myosin binding sites. Option B is incorrect because acetylcholine release at the neuromuscular junction is the event that initiates muscle contraction, not relaxation. Additional acetylcholine release would stimulate another contraction, rather than causing the muscle to relax. Option C is incorrect because sodium, entering through the muscle fiber membrane, is part of the depolarization process that leads to contraction. It is an excitatory event that occurs before calcium release, not a relaxation mechanism. Option D is incorrect because troponin binding to additional calcium would keep the myosin binding sites on actin exposed and maintain cross-bridge cycling. This would continue contraction, not initiate the relaxation of the muscle. Relaxation is the reverse of contraction, and it starts when calcium gets pumped back into the sarcoplasmic reticulum. Once calcium is removed from the cytoplasm, troponin returns to its original shape, tropomyosin slides back over the myosin binding sites on actin, and cross-bridge cycling stops. The muscle relaxes. UC release at the neuromuscular junction is what starts a contraction. More EC would trigger another one, not relaxation. Sodium entering through the membrane is part of depolarization, which leads to contraction. And troponin binding more calcium would keep the binding sites exposed, which means the muscle keeps contracting. The only event that kicks off relaxation is calcium being actively pumped back into storage. Here's a WGU D312 OA exam tip for you. Relaxation equals calcium removal. If a question asks what directly causes relaxation, look for calcium pumped back into the sarcoplasmic reticulum. Quick hook. No calcium, no contraction. Pump it back, muscle relaxes. 15. After a single muscle contraction event at the neuromuscular junction, the signaling molecule must be rapidly removed from the synaptic cleft to prevent continuous and uncontrolled stimulation of the muscle fiber. Which enzyme is responsible for breaking down and clearing this specific signaling molecule? A. Troponin located on the thin filaments. B. Calcium atpase in the reticulum. C. Myosin atpase on the thick filaments. D. Acetylcholinesterase in the synaptic cleft. Can you guess the answer? Correct answer. D. Acetylcholinesterase in the synaptic cleft. Explanation. Option D is correct because acetylcholinesterase is the enzyme located in the synaptic cleft that rapidly breaks down acetylcholine after it has bound to receptors. This prevents continuous stimulation of the muscle fiber membrane. Option A is incorrect because troponin is a regulatory protein found on the thin actin filaments inside the muscle fiber. It is not an enzyme and has no role in breaking down neurotransmitters in the synaptic cleft. Option B is incorrect because calcium atpase is a pump enzyme in the sarcoplasmic reticulum membrane that moves calcium back into storage during relaxation. It does not act in the synaptic cleft to remove neurotransmitters. Option C is incorrect because myosin atpase is an enzyme on the myosin heads that breaks down ATP to provide energy for the power stroke during cross-bridge cycling. It operates inside the muscle fiber, not in the synapse. After a muscle contraction event, acetylcholine is sitting in the synaptic cleft still bound to receptors. If nothing clears it away, the muscle fiber would keep getting stimulated non-stop, uncontrolled twitching. The enzyme that prevents this is acetylcholinesterase. It lives right there in the synaptic cleft and rapidly breaks AC into inactive fragments so the signal stops cleanly. Troponin is a regulatory protein on actin filaments inside the fiber. It's not an enzyme and it doesn't touch the synapse. Calcium atpase is a pump in the sarcoplasmic reticulum membrane that pulls calcium back into storage during relaxation. Totally different location and function. Myosin atpase sits on the myosin heads and breaks down ATP for the power stroke during cross-bridge cycling. Again inside the fiber, not in the cleft. Here's a WGU D312 OA exam tip for you. When a question asks what clears AC from the synaptic cleft, it's acetylcholinesterase, always. The name tells you everything. Acetylcholinesterase breaks down acetylcholine. Quick hook. HE equals sign UC eraser. 16. A patient experiences difficulty with involuntary processes such as regulating heart rate, controlling digestive activity, and managing blood vessel diameter. These functions are controlled by a specific division of the peripheral nervous system that operates without conscious effort. Which division is most directly responsible? A. The somatic nervous system division. B. the autonomic nervous system division? C, the enteric nervous system only? D, the central nervous system alone? What do you think is the answer? Correct answer, B, the autonomic nervous system division. Explanation. Option B is correct because the autonomic nervous system controls involuntary functions, including heart rate, blood pressure, digestion, and blood vessel diameter. It operates through sympathetic and parasympathetic subdivisions without conscious control. Option A is incorrect because the somatic nervous system handles voluntary motor control of skeletal muscles and receives sensory input from the body surface. It does not control involuntary visceral functions like heart rate or digestion. Option C is incorrect because the enteric nervous system specifically creates nerve plexuses that innervate the gastrointestinal tract. While involved in digestion, it does not directly control heart rate, blood pressure, or vessel diameter. Option D is incorrect because the central nervous system consists of the brain and spinal cord and serves as the processing center. Involuntary regulation of organs is carried out by the autonomic division of the peripheral system. The patient can't control heart rate, digestion, or blood vessel diameter. All involuntary processes running without conscious effort. That's the autonomic nervous system. The ANS is a division of the peripheral nervous system that regulates smooth muscle, cardiac muscle, and glands. It splits into the sympathetic branch for fight or flight, and the parasympathetic branch for rest and digest. The somatic nervous system is voluntary. It controls skeletal muscles and receives sensory input from the body surface. It doesn't run your heart or your gut. The enteric nervous system does handle the gastrointestinal tract, but it's limited to digestion. It doesn't regulate heart rate or blood vessel diameter. The central nervous system is the brain and spinal cord. It processes information, but the actual regulation of organs is carried out through the autonomic division of the PNS. Here's a WGU D312 OA exam tip for you. Involuntary control of organs, glands, heart rate, and digestion points to the autonomic nervous system. Voluntary skeletal muscle control is somatic. Quick hook. Autonomic = automatic. Runs without you thinking about it. 17. In the central nervous system, a specific type of glial cell is responsible for wrapping axons in a myelin sheath to provide insulation and speed up nerve impulse conduction along the neural pathways. Which type of glial cell performs this myelination function within the central nervous system specifically? A. Oligodendrocytes in the central system. B. Schwann cells in the peripheral nerves. C. Astrocytes providing structural support. D. Microglia performing immune surveillance. What do you think is the answer? Correct answer, A. Oligodendrocytes in the central system. Explanation. Option A is correct because oligodendrocytes are the glial cells responsible for producing myelin sheaths around axons in the central nervous system. They insulate axons to increase the speed of nerve impulse conduction throughout the brain and cord. Option B is incorrect because Schwann cells produce myelin in the peripheral nervous system, not the central nervous system. While they perform the same myelination function, their location is restricted to nerves outside the brain and cord. Option C is incorrect because astrocytes are central nervous system glial cells that provide structural support and help maintain the blood-brain barrier. They do not produce myelin sheaths or insulate axons within the neural pathways. Option D is incorrect because microglia are the immune cells of the central nervous system that perform phagocytosis and immune surveillance. They protect against pathogens and debris, but do not produce myelin around axons. Which glial cell wraps axons in myelin inside the central nervous system? Oligodendrocytes. They're the CNS myelinating cells. Myelin is the fatty insulation sheath that speeds up nerve impulse conduction by allowing signals to jump between gaps, saltatory conduction. Schwann cells do the same job, but only in the peripheral nervous system, outside the brain and spinal cord. That's the key distinction the exam loves to test. Astrocytes provide structural support and help maintain the blood-brain barrier, but they don't make myelin. Microglia are the immune cells of the CNS. They perform phagocytosis to clean up pathogens and debris. Neither of those has anything to do with myelination. Here's a WGU D312 OA exam tip for you. CNS myelin equals oligodendrocytes. PNS myelin equals Schwann cells. If a question specifies the central nervous system, don't pick Schwann cells. Quick hook. Oligo equals sign CNS, Schwann equals sign PNS. Or think O for oligo and O for only in the CNS. 18. A physician explains to a medical student that the brain and spinal cord are protected by 3 membrane layers. The outermost layer is the toughest and most durable, while the innermost layer is thin and permeable, sitting directly on the brain surface. Which layer is this innermost protective membrane? A. The dura mater as the tough outer layer. B. The arachnoid mater in the middle position. C. The periosteum covering the skull bones. D. The pia mater as the innermost layer. Do you know the answer? Correct answer, D. The pia mater as the innermost layer. Explanation. Option D is correct because the pia mater is the innermost meningeal layer, a thin fibrous tissue sitting directly on the brain surface. It is permeable to water and small solids, allowing blood vessels to nourish the brain. Option A is incorrect because the dura mater is the outermost meningeal layer, not the innermost. It is the toughest and most durable of the 3 membranes protecting the brain and spinal cord from external forces. Option B is incorrect because the arachnoid mater is the middle meningeal layer, positioned between the dura mater and the pia mater. It forms a loosely fitting sac and is not the membrane directly contacting the brain surface. Option C is incorrect because the periosteum is the fibrous membrane covering the outer surface of bones throughout the body. It is not one of the 3 meningeal layers and does not directly protect the brain tissue. 3 membrane layers protect the brain and spinal cord. The meninges, from outside in, dura mater, arachnoid mater, pia mater. The question asks about the innermost layer that sits directly on the brain surface and is thin and permeable. That's the pia mater. It's a delicate fibrous tissue that lets water and small solutes pass through so blood vessels can penetrate it and feed the brain. The dura mater is the outermost layer, tough and thick, the opposite of what the question describes. The arachnoid mater sits in the middle, forming a loose sac between the dura and the pia. The periosteum covers bone surfaces, not brain tissue. It's not even one of the meninges. Here's a WGU D312 OA exam tip for you. Memorize the meninges from outside in. Dura, arachnoid, pia. The innermost is always the pia mater. Quick hook, DAP from outside in. Or, the pia is plastered right on the brain. 19. A physician performing a lumbar puncture on an adult patient inserts a needle below the point where the spinal cord tapers and terminates. Below this termination point, only a bundle of nerve roots from the lumbar and sacral regions continues downward. Which anatomical structure describes this descending nerve bundle? A. The conus medullaris at its terminus. B. The filum terminale extension below. C. The cauda equina nerve root bundle. D. The central canal of the spinal cord. What do you think is the answer? Correct answer, C. The cauda equina nerve root bundle. Explanation. Option C is correct because the cauda equina is the bundle of lumbar, sacral, and coccygeal nerve roots that extends below the conus medullaris where the spinal cord terminates. Its name means horsetail due to its appearance. Option A is incorrect because the conus medullaris is the tapered lower end of the spinal cord itself, located near the first lumbar vertebral level. It is the termination point of the cord, not the nerve bundle below it. Option B is incorrect because the filum terminale is an extension of the pia mater that anchors the spinal cord to the coccygeal segments below. It is a structural tether, not a bundle of functional nerve roots. Option D is incorrect because the central canal is a narrow channel running through the centre of the spinal cord that contains cerebrospinal fluid. It does not extend below the cord as a bundle of nerve roots. A lumbar puncture needle goes in below where the spinal cord actually ends. The cord tapers off at the conus medullaris, around the first lumbar vertebra in adults. Below that point, a bundle of nerve roots, lumbar, sacral, and coccygeal, dangles downward through the vertebral canal. That bundle is the cauda equina, Latin for horse's tail, because that's exactly what it looks like. The conus medullaris is the tapered end of the cord itself, not the nerve bundle below it. The filum terminale is a strand of pia mater that anchors the cord to the coccyx. It's a tether, not functional nerve roots. The central canal runs through the middle of the spinal cord and carries cerebrospinal fluid. It doesn't continue below the cord as a nerve bundle. Here's a WGU D312 OA exam tip for you. Nerve roots dangling below the end of the spinal cord equals cauda equina. If a question mentions lumbar puncture below the cord termination, think cauda equina, quick hook. Cauda equina equals sign horsetail equals sign nerve roots hanging below the cord. 20. A neuroscience student is studying neurons that transmit sensory information from receptors in the skin and joints toward the brain and spinal cord for processing and interpretation. These neurons carry impulses in one specific direction. Which functional classification best describes these particular neurons? A. Motor efferent neurons leaving the central nervous system. B. Sensory afferent neurons entering the central nervous system. C. Interneurons found within the spinal cord. D. Multipolar neurons with many dendrites. Can you guess the answer? Correct answer, B. Sensory afferent neurons entering the central nervous system. Explanation. Option B is correct because sensory afferent neurons transmit information from receptors in the body toward the central nervous system for processing. They carry impulses from the periphery inward to the brain and spinal cord. Option A is incorrect because motor efferent neurons carry impulses in the opposite direction, away from the central nervous system toward effector organs like muscles and glands. They do not transmit sensory information inward. Option C is incorrect because interneurons are located entirely within the central nervous system and serve as bridges between sensory and motor neurons. They do not extend out to skin receptors or peripheral body structures. Option D is incorrect because multipolar is a morphological classification based on the number of processes extending from the cell body. It describes structure, not the functional direction of impulse transmission toward or away from the central nervous system. Neurons that carry sensory information from receptors in the skin and joints toward the brain and spinal cord are called sensory afferent neurons. The word afferent means toward the centre, so afferent neurons bring information into the CNS for processing. Motor efferent neurons go the other direction. They carry impulses away from the CNS out to muscles and glands. Interneurons sit entirely within the CNS and bridge the gap between sensory and motor neurons. They don't reach out to skin receptors. Multipolar is a structural classification describing neurons with multiple dendrites and one axon. It tells you about shape, not about the direction of signal travel. The question asks about functional classification, direction, not morphology. Here's a WGU D312 OA exam tip for you. Toward the CNS equals afferent. Away from the CNS equals efferent. If a question describes sensory signals travelling inward to the brain, that's afferent. Quick hook. Afferent arrives at the CNS. Efferent exits the CNS. 21. A water-soluble hormone arrives at its target cell but cannot cross through the plasma membrane due to its chemical properties. Instead, it binds to a receptor on the outer surface and triggers an internal cascade of molecular events. How does this hormone relay its message to produce a cellular response? A. Through a second messenger system. B. By entering the nucleus directly. C. By dissolving through the membrane. D. Through direct gene activation inside. Do you know the answer? Correct answer, A. Through a second messenger system. Explanation. Option A is correct because water-soluble hormones cannot cross the cell membrane, so they bind to surface receptors and use intracellular second messenger molecules to relay the signal from the receptor to targets within the cell. Option B is incorrect because water-soluble hormones cannot pass through the lipid-based cell membrane to reach the nucleus directly. Only lipid-soluble hormones can dissolve through the membrane and interact with intracellular or nuclear receptors. Option C is incorrect because water-soluble hormones are unable to dissolve through the hydrophobic lipid bilayer of the cell membrane. Their chemical properties prevent them from crossing, which is exactly why they require surface receptors instead. Option D is incorrect because direct gene activation inside the nucleus is the mechanism used by lipid-soluble hormones that can enter the cell. Water-soluble hormones affect the cell through surface receptor binding and second messenger cascades. This one is about how water-soluble hormones get their message across. A water-soluble hormone can't pass through the cell membrane. The membrane is made of lipids and water-soluble molecules don't dissolve in fat. So the hormone stays outside, binds to a receptor on the cell surface, and that receptor triggers an internal cascade using what's called a second messenger system. The second messenger is an intracellular molecule that relays the signal from the surface receptor to targets deeper in the cell, entering the nucleus directly. That's what lipid-soluble hormones do. They slip through the membrane because they dissolve in fat. Dissolving through the membrane is the same problem restated. Water-soluble hormones simply can't do that. Direct gene activation is also a lipid-soluble hormone move, where the hormone receptor complex binds DNA in the nucleus and changes gene expression. Here's a WGU D312 OA exam tip for you. Water-soluble equals stays outside, uses second messenger. Lipid-soluble equals enters the cell, changes genes directly. Quick hook, water stays out, uses a messenger. Lipid goes in, talks to genes. 22. A lipid-soluble hormone formed from cholesterol is released into the bloodstream and travels to its target cell. Unlike water-soluble hormones, it passes directly through the cell membrane and binds to an intracellular receptor that forms a complex. What happens after this hormone receptor complex forms inside the target cell? A. A second messenger activates outside. B. The hormone stays on the surface. C. Gene expression changes in the nucleus. D. The cell membrane receptor depolarizes. Do you know the answer? Correct answer, C. Gene expression changes in the nucleus. Explanation. Option C is correct because when a lipid-soluble hormone binds its intracellular receptor, the hormone receptor complex moves to the nucleus and directly alters gene expression. Translation then produces new proteins from the genetic code carried by messenger RNA. Option A is incorrect because second messenger systems are used by water-soluble hormones that bind surface receptors. Lipid-soluble hormones bypass the need for second messengers by entering the cell and binding intracellular receptors directly inside. Option B is incorrect because lipid-soluble hormones do not remain on the cell surface. Their fat-soluble nature allows them to dissolve through the plasma membrane and enter the cell, where they bind to receptors in the cytoplasm or nucleus. Option D is incorrect because depolarization of a cell membrane is a process associated with nerve impulse transmission and neuromuscular signaling. It is not the mechanism by which lipid-soluble hormones produce their effects inside target cells. Now we flip to lipid-soluble hormones. These are usually made from cholesterol, so they dissolve right through the fatty cell membrane. Once inside, they bind an intracellular receptor, and that hormone receptor complex heads to the nucleus. There, it directly changes gene expression, turning genes on or off. Translation then produces new proteins from the mRNA code. That's the whole mechanism. Cross the membrane, bind an intracellular receptor, alter gene expression. A second messenger activating outside is the water-soluble pathway, surface receptors and cascades, not intracellular binding. Staying on the surface is wrong because lipid-soluble hormones pass right through. Membrane depolarization is a nerve and muscle thing. Action potentials, not hormone signaling. Here's a WGU D312 OA exam tip for you. When a question says a hormone crosses the membrane and binds inside the cell, the result is gene expression change. That's the lipid-soluble pathway. Quick hook. Lipid equals sign, lipo equals sign, fat equals sign, floats through fat membrane equals sign gene change. 23. The anterior pituitary gland contains 5 types of specialized endocrine cells, each producing a different hormone that targets specific organs or tissues throughout the body. Which cell type in the anterior pituitary gland is specifically responsible for producing thyroid-stimulating hormone that acts on the thyroid? A. Somatotrophs producing growth hormone. B. Lactotrophs producing prolactin. C. Corticotrophs producing ACT. D. Thyrotrophs producing thyroid-stimulating hormone. Do you know the answer? Correct answer, D. Thyrotrophs producing thyroid-stimulating hormone. Explanation. Option D is correct because thyrotrophs are the specific anterior pituitary endocrine cells that produce thyroid-stimulating hormone, also known as TSH. This hormone travels to the thyroid gland and stimulates synthesis of thyroid hormones. Option A is incorrect because somatotrophs are anterior pituitary cells that produce human growth hormone, which targets the liver, skeletal muscle, cartilage, and bone. They do not produce thyroid-stimulating hormone. Option B is incorrect because lactotrophs are anterior pituitary cells that produce prolactin, which targets the mammary glands to stimulate milk production. They have no role in thyroid gland stimulation or thyroid hormone synthesis. Option C is incorrect because corticotrophs are anterior pituitary cells that produce adrenocorticotropic hormone, which targets the adrenal cortex. They stimulate cortisol production, but have no direct effect on the thyroid gland. The anterior pituitary has 5 types of endocrine cells, and this question asks which one makes TSH, thyroid-stimulating hormone. That's the thyrotroph. The name gives it away. Thyrotroph feeds the thyroid. TSH travels to the thyroid gland and tells it to make T3 and T4. Somatotrophs produce growth hormone targeting the liver, bone, muscle, and cartilage. Lactotrophs produce prolactin for the mammary glands. Corticotrophs produce ACTH, which targets the adrenal cortex for cortisol production. Each cell type has one job, one hormone, one target system. Here's a WGU D312 OA exam tip for you. Match the prefix to the target. Thyro equals sign thyroid. Somato equals sign body, growth. Lacto equals sign milk. Cortico equals sign cortex. Gonado equals sign gonads. If the question asks about TSH, look for thyrotroph. Quick hook. Thyrotroph feeds the thyroid. 24. The hypothalamus controls anterior pituitary hormone secretion by sending regulatory hormones through a specialized portal blood vessel system. Some regulatory hormones stimulate release, while others inhibit it. Which hypothalamic hormone directly stimulates the anterior pituitary somatotrophs to secrete growth hormone? A. GHIH that inhibits growth hormone. B. GRH that stimulates growth hormone. C. CRH that targets the corticotrophs. D. TRH that targets the thyrotrophs. What do you think is the answer? Correct answer, B. GHRH, that stimulates growth hormone. Explanation. Option B is correct, because growth hormone-releasing hormone from the hypothalamus travels through the portal system and directly stimulates somatotrophs in the anterior pituitary to secrete human growth hormone into the bloodstream. Option A is incorrect because growth hormone inhibiting hormone has the opposite effect and suppresses growth hormone secretion from somatotrophs. It is an inhibitory signal from the hypothalamus, not a stimulatory one targeting growth hormone release. Option C is incorrect because corticotropin, releasing hormone from the hypothalamus, targets corticotrophs in the anterior pituitary to stimulate ACEH secretion. It acts on a completely different cell type and hormone pathway than growth hormone. Option D is incorrect because thyrotropin releasing hormone from the hypothalamus targets thyrotrophs in the anterior pituitary to stimulate TSH secretion. It does not act on somatotrophs and has no direct role in growth hormone release. The hypothalamus sends regulatory hormones down through a portal blood system to control what the anterior pituitary releases. This question asks which hypothalamic hormone stimulates somatotrophs to secrete growth hormone. That's GHRH, growth hormone releasing hormone. The name spells it out. GIH is growth hormone inhibiting hormone. It does the opposite, suppressing growth hormone release from somatotrophs. CRH is corticotropin releasing hormone, which targets corticotrophs for ACT, completely different pathway. TRH is thyrotropin releasing hormone, which targets thyrotrophs for TSH. Each releasing hormone has a specific match in the anterior pituitary. Here's a WGU D312 OA exam tip for you. If the question says stimulates growth hormone, that's GHRH. If it says inhibits, that's GH. The R in GH stands for releasing. Quick hook, GAR equals sign go hormone, release him. GA equals sign go hormone, inhibit him. 25. Unlike the anterior pituitary, the posterior pituitary does not synthesize its own hormones, but stores and releases hormones made by hypothalamic neurons. One specific hypothalamic nucleus produces both oxytocin and antidiuretic hormone for release from the posterior pituitary. Which nucleus synthesizes both of these hormones? A. The paraventricular nucleus. B. The anterior pituitary gland cells. C. The supraoptic nucleus exclusively. D. The median eminence portal region. Do you know the answer? Correct answer, A. The paraventricular nucleus. Explanation. Option A is correct because the paraventricular nucleus of the hypothalamus synthesizes both oxytocin and antidiuretic. Hormone. Its magnocellular neurons send axons to the posterior pituitary, where these hormones are stored and released into circulation. Option B is incorrect because the anterior pituitary gland cells produce their own distinct set of hormones such as growth hormone and TSH. They do not synthesize oxytocin or antidiuretic hormone, which come from the hypothalamus. Option C is incorrect because the supraoptic nucleus primarily synthesizes and secretes antidiuretic hormone, but is not identified as the source of both hormones. The paraventricular nucleus is the one that produces both oxytocin and antidiuretic hormone. Option D is incorrect because the median eminence is the region where hypothalamic releasing hormones enter the portal blood system to reach the anterior pituitary. It is not a nucleus that synthesizes posterior pituitary hormones. The posterior pituitary doesn't make its own hormones, it just stores and releases them. The hormones come from neurons in the hypothalamus. This question asks which nucleus makes both oxytocin and ADH. That's the paraventricular nucleus, PVN. Its magnocellular neuron synthesize both hormones, and the axons run down to the posterior pituitary where the hormones sit until they're needed. The supraoptic nucleus mainly produces ADH, but the question specifically asks for the nucleus that makes both. The anterior pituitary cells make a completely different set of hormones. Growth hormone, TSH, ASTH, prolactin, FSH, LH, none of which are oxytocin or ADH. The median eminence is just the portal system gateway for releasing hormones headed to the anterior pituitary. It's not a nucleus at all. Here's a WGU D312 OA exam tip for you. PVN makes both oxytocin and ADH. Sun makes mainly ADH. If the question says both hormones, pick the paraventricular nucleus. Quick hook. PVN equals sign pair of hormones, oxytocin and ADH from the same nucleus. 26. A patient arrives at the emergency department after a head injury and is unable to detect any odours during a clinical smell test, even when strong scents are placed directly beneath the nose. Which cranial nerve has most likely been damaged to produce this specific sensory deficit? A. CN2 responsible for vision. B. CN7 for facial expression. C. CN for taste and swallowing. D. CNI responsible for smell. What do you think is the answer? Correct answer. D. CNI responsible for smell. Explanation. Option D is correct because cranial nerve 1, the olfactory nerve, is solely responsible for the sense of smell. Damage to this nerve eliminates the ability to detect odours, which matches the clinical presentation described in this scenario. Option A is incorrect because cranial nerve 2 is the optic nerve, which carries visual impulses from the retina to the brain. Damage to this nerve would affect vision, not the ability to detect odours during a clinical smell test. Option B is incorrect because cranial nerve 7 is the facial nerve, which controls muscles of facial expression, some taste sensation, and swallowing. It does not carry olfactory information from the nasal cavity to the brain. Option C is incorrect because cranial nerve 9 is the glossopharyngeal nerve, which is involved in taste from the posterior tongue, swallowing, and pharyngeal sensation. It plays no role in detecting airborne odours through the nose. Head injury, no ability to smell anything, even strong scents right under the nose. The damaged nerve is CNY, the olfactory nerve. It's the only cranial nerve responsible for smell. CN2 is the optic nerve for vision. Damage there causes visual problems, not smell loss. CN7 is the facial nerve for facial expressions and some taste. It has nothing to do with detecting odours through the nose. CN9 is the glossopharyngeal nerve for posterior tongue taste and swallowing. Again, zero involvement in olfaction. The cranial nerves each have specific jobs, and smell belongs exclusively to CNY. Here's a WGU D312 OA exam tip for you. Loss of smell after head injury equals CNI damage. The olfactory nerve fibres pass through the cribriform plate, which is fragile and vulnerable during head trauma. Quick hook. CNI equals sign, nose equals sign. Number one thing you smell. 27. A patient presents with an inability to move one eye laterally, meaning the affected eye cannot look outward toward the ear on that same side. The physician suspects damage to a single cranial nerve that exclusively controls the lateral rectus muscle. Which cranial nerve is most likely injured? A. CN6, the abducens nerve. B. CN3, the oculomotor nerve. C. CNIV, the trochlear nerve. D. CNV, the trigeminal nerve. Do you know the answer? Correct answer. A. CNV, the abducens nerve. Explanation. Option A is correct because cranial nerve 6, the abducens nerve, exclusively innervates the lateral rectus muscle that abducts the eye. Damage to this nerve prevents the eye from looking outward toward the ear on the affected side. Option B is incorrect because cranial nerve 3, the oculomotor nerve, controls the superior rectus, inferior rectus, medial rectus, and inferior oblique muscles. It moves the eye in several directions, but does not control lateral abduction. Option C is incorrect because cranial nerve 4, the trochlear nerve, innervates only the superior oblique muscle. This muscle depresses the cornea, using the trochlear as a pulley, and is not responsible for lateral eye movement. Option D is incorrect because cranial nerve 5, the trigeminal nerve, provides sensory information from the face and head and controls the muscles of mastication. It has no motor function related to eye movement in any direction. The patient can't move one eye outward, can't look laterally toward the ear. The muscle that abducts the eye is the lateral rectus, and the only nerve supplying it is CN-VIX, the abducens nerve. The name gives you the answer. Abducens equals sign abduction, equals sign moving the eye outward. CN3, the oculomotor nerve, controls most of the other eye muscles. Superior rectus, inferior rectus, medial rectus, inferior oblique. It handles up, down and inward movement, but not outward. CN4, the trochlear nerve, innervates only the superior oblique, which depresses and rotates the eye, not lateral movement. CNV, the trigeminal nerve, handles face sensation and chewing muscles. It has no motor function related to eye movement whatsoever. Here's a WGU D312 OA exam tip for you. Can't look laterally equals lateral rectus equals CN-VIC abducens. This is one of the most commonly tested cranial nerve facts. Quick hook. CN-VIC equals sign 6 equals sign lateral rectus equals sign looks to the side. 28. During a neurological examination, a physician asks the patient to clench their jaw tightly and then test sensation across the forehead, cheek, and chin. Both the motor function of jaw clenching and the sensory input from these facial regions are carried by a single cranial nerve. Which nerve serves both functions? A. CN7, the facial nerve. B. CN-KEX20, the hypoglossal nerve. C. CNV, the trigeminal nerve. D. CN-CAM10, the accessory nerve. What do you think is the answer? Correct answer, C. CNV, the trigeminal nerve. Explanation. Option C is correct because cranial nerve 5, the trigeminal nerve, has both sensory and motor functions. It provides sensation across the face and head, and its motor component controls the muscles of mastication, including jaw clenching. Option A is incorrect because cranial nerve 7, the facial nerve, controls the muscles of facial expression rather than jaw clenching. While it carries some taste and sensory information, it does not provide general sensation across the forehead and chin. Option B is incorrect because cranial nerve 12, the hypoglossal nerve, controls the muscles of the tongue. It has no role in jaw clenching or providing sensory input from the forehead, cheek, or chin regions of the face. Option D is incorrect because cranial nerve 11, the accessory nerve, controls muscles involved in head movement, such as the sternocleidomastoid and trapezius. It does not provide facial sensation or control the muscles that clench the jaw. Jaw clenching plus sensation from the forehead, cheek and chin, all handled by one nerve, that's CNV, the trigeminal nerve. It's called trigeminal because it has 3 branches covering 3 zones of the face. Ophthalmic for the forehead, maxillary for the cheek, mandibular for the chin and jaw. The motor component of the mandibular branch drives the muscles of mastication. That's the jaw clench. CN7, the facial nerve, controls facial expressions like smiling and frowning, plus some taste, but it doesn't run the chewing muscles or provide general sensation across the face. CN-KEX20, the hypoglossal nerve, moves the tongue. CN-KEX20, the accessory nerve, controls the sternocleidomastoid and trapezius for head and shoulder movement. Neither of those touches facial sensation or jaw muscles. Here's a WGU D312 OA exam tip for you. Jaw clenching plus face sensation in one question equals trigeminal, CNV. Remember it this way. The trigeminal handles TRI things, forehead, cheek, chin. Quick hook, trigeminal equals sign 3 zones of the face plus jaw. 29. A clinician evaluates a patient who has lost the ability to taste food on the posterior third of the tongue and also has difficulty swallowing. The physician suspects a single cranial nerve lesion affecting both taste reception from that tongue region and the swallowing reflex. Which cranial nerve is most likely affected? A. CN7, the facial nerve. B. CN9, the glossopharyngeal nerve. C. CN-KEX20, the hypoglossal nerve. D. CNX, the vagus nerve. What do you think is the answer? Correct answer, B. CNIX, the glossopharyngeal nerve. Explanation. Option B is correct because cranial nerve 9, the glossopharyngeal nerve, receives taste input from the valate and foliate papillae on the posterior tongue and also contributes to the swallowing reflex. Damage explains both symptoms simultaneously. Option A is incorrect because cranial nerve 7, the facial nerve, is associated with taste from the anterior tongue and innervates taste buds in the epiglottis and throat. It does not receive primary taste input from the posterior third of the tongue. Option C is incorrect because cranial nerve 12, the hypoglossal nerve, controls tongue muscle movements needed for speech and manipulating food. It does not carry taste information from any region of the tongue or directly control swallowing. Option D is incorrect because cranial nerve 10, the vagus nerve, has broad autonomic visceral functions and receives taste input from the fungiform papillae. While involved in swallowing, it is not the primary nerve for posterior tongue taste. Lost taste on the posterior third of the tongue and difficulty swallowing, both from one cranial nerve lesion. That's CN9, the glossopharyngeal nerve. The name breaks down. Glosso means tongue, pharyngeal means throat. CN9 handles taste from the valate and foliate papillae on the back of the tongue and contributes to the swallowing reflex. CN7, the facial nerve, deals with taste from the anterior tongue and innervates taste buds in the epiglottis and throat, not the posterior third specifically. CN12, the hypoglossal nerve, controls tongue muscle movement for speech and chewing, but it carries zero taste information. CNX, the vagus nerve, has wide autonomic functions and receives taste from fungiform papillae. But it's not the primary nerve for posterior tongue taste. Here's a WGUD 312 OA exam tip for you. Posterior tongue taste plus swallowing problems equals CN9. Glossopharyngeal, quick hook. Glosso equals sign tongue, pharyngeal equals sign throat. CN9 does both. 30. A patient cannot move their tongue properly, resulting in slurred speech and difficulty positioning food during chewing. The physician determines that the cranial nerve responsible for controlling tongue muscle movement has been damaged on one side. Which cranial nerve controls tongue muscle movement? A. CN9, the glossopharyngeal nerve. B. CN7, the facial nerve. C. CNX, the vagus nerve. D. CN12, the hypoglossal nerve. What do you think is the answer? Correct answer, D. CN12, the hypoglossal nerve. Explanation. Option D is correct because cranial nerve 12, the hypoglossal nerve, is specifically responsible for controlling the muscles of the tongue. Damage to this nerve impairs tongue movement, causing difficulty with speech and food positioning. Option A is incorrect because cranial nerve 9, the glossopharyngeal nerve, is involved in taste sensation from the posterior tongue and swallowing. It does not provide motor control to the muscles that move the tongue during speech and chewing. Option B is incorrect because cranial nerve 7, the facial nerve, controls the muscles of facial expression such as smiling, frowning, and closing the eyes. It does not innervate the muscles responsible for tongue movement. Option C is incorrect because cranial nerve 10, the vagus nerve, has widespread autonomic and sensory functions involving the thoracic and abdominal organs. It does not directly control the voluntary muscle movements of the tongue. The patient can't move their tongue properly, slurred speech, trouble positioning food. The nerve that controls tongue muscles is CN12, the hypoglossal nerve. Hypo means under, glossal means tongue. The nerve that runs under the tongue to move it. CN9, the glossopharyngeal, handles posterior tongue taste and swallowing, but not tongue muscle movement. CN7, the facial nerve, controls the muscles of facial expression, smiling, frowning, blinking, not the tongue. CNX, the vagus nerve, runs through the chest and abdomen, handling autonomic functions for organs. it doesn't directly control the voluntary muscles that move the tongue during speech. Here's a WGU D312 OA exam tip for you. Tongue movement problems equals CN Kickspower hypoglossal. If a question says slurred speech or difficulty moving the tongue, that's your answer. Quick hook. Hypoglossal equals sign under the tongue equals sign moves the tongue. 12 muscles working under there. 31. A physician shines a light into a patient's eye and explains to a medical student that the light must pass through the transparent front dome of the eye, then through a fluid-filled space, then through the adjustable lens before reaching the light-sensitive layer at the back. Which layer at the back of the eye contains the photoreceptors that detect the light? A. The retina as the neural tunica. B. The choroid as the vascular layer. C. The sclera as the outer fibrous layer. D. The cornea at the front of the eye. What do you think is the answer? Correct answer. A. The retina as the neural tunica. Explanation. Option A is correct because the retina is the innermost neural tunica that lines the back of the eye. It contains photoreceptor cells called rods and cones that detect light and convert optical images into neural signals for the brain. Option B is incorrect because the choroid is the middle vascular tunica of the eye that provides blood supply and drains excess fluid from the posterior cavity. It does not contain photoreceptor cells that detect and respond to light. Option C is incorrect because the sclera is the outermost fibrous tunica that forms the white of the eye. It maintains the eye shape and prevents light from entering except through the cornea, but it does not detect light. Option D is incorrect because the cornea is the transparent dome at the front of the eye that refracts light inward. While it is the first structure light passes through, it does not contain photoreceptor cells that convert light into neural signals. Light passes through the front of the eye, through fluid, through the lens, and lands on a light-sensitive layer at the back. That layer is the retina, the neural tunica, the innermost of the eye's 3 layers. The retina contains photoreceptors, rods for dim light and cones for colour. It converts optical images into neural signals that travel via the optics nerve to the brain. The choroid is the middle layer, the vascular tunica, packed with blood vessels that supply nutrients and drain fluid. It doesn't detect light. The sclera is the outer layer, the white of the eye. It maintains shape and blocks stray light, but it has no photoreceptors. The cornea is the transparent dome at the front that refracts light inward. It's the first structure light hits, not the detecting layer at the back. Here's a WGU D312 OA exam tip for you. The 3 tunicae from outside in are fibrous, sclera cornea, vascular, choroid, ciliary, body, iris, and neural, retina. Photoreceptors live in the retina. Quick hook. Retina equals sign receives light and reports to the brain. 32. A patient reports difficulty seeing in dimly lit environments but has no trouble identifying colours in bright daylight. An ophthalmologist suspects a problem with a specific type of photoreceptor that functions best in low-light conditions and provides vision without colour. Which photoreceptor type is most likely affected? A. Cone cells sensitive to bright light. B. Ganglion cells at the optic disc. C. Rod cells sensitive to low light. D. Bipolar cells in the retinal layers. What do you think is the answer? Correct answer, C. Rod cells sensitive to low light. Explanation. Option C is correct because rod cells are photoreceptors sensitive to low light levels that provide black and white vision. They contain the photopigment rhodopsin and are essential for seeing in dimly lit environments without colour discrimination. Option A is incorrect because cone cells are photoreceptors sensitive to highlight levels that provide colour vision. Since the patient can see colours well in bright daylight, the cone cells are functioning properly and are not the affected type. Option B is incorrect because ganglion cells are retinal neurons whose axons form the optic nerve. They relay processed visual signals from the retina to the brain, but are not the primary photoreceptor cells that detect dim light. Option D is incorrect because bipolar cells are intermediate neurons in the retina that connect photoreceptors to ganglion cells. They transmit signals within the retinal processing chain, but do not themselves detect light in dim conditions. The patient sees fine in bright light and recognizes colors perfectly, but struggles in dim environments. That points to a problem with rod cells. Rods are the photoreceptors built for low light vision. They contain rhodopsin and give you black and white vision when the lights are low. Cones handle bright light and color. Since this patient sees colors fine in daylight, cones are working. Ganglion cells are retinal neurons whose axons form the optic nerve. They relay processed signals but aren't the primary light detectors. Bipolar cells sit between photoreceptors and ganglion cells in the retinal processing chain. They're intermediaries, not the cells that actually sense dim light. Here's a WGU D312 OA exam tip for you. Dim light problems equal rod cells. Bright light and colour problems equal cone cells. If a patient can see colours but not in the dark, rods are the issue. Quick hook. Rods equal sign dim room vision. Cones equal sign colour vision. 33. An ophthalmologist examines a patient's eye and identifies the region where the clearest central vision occurs, noting that this small area at the centre of the macula contains only cone photoreceptors and no rods. The physician explains this is where focused images are projected for maximum visual acuity. Which structure is being described? A. The optic disc where axons exit. B. The ora serrata at the retina edge. C. The macula lutea surrounding the area. D. The fovea centralis at the macula centre. Do you know the answer? Correct answer. D. The fovea centralis at the macula centre. Explanation. Option D is correct because the fovea centralis is the pinpoint depression at the centre of the macula lutea, where vision is most accurate. It contains only cone photoreceptors, providing the highest visual acuity for detailed and colour vision. Option A is incorrect because the optic disc is the point where ganglion cell axons exit the eye to form the optic nerve. It is the blind spot because it contains no photoreceptors at all, making it unable to produce any visual image. Option B is incorrect because the ora serrata is the border where the retina ends at the anterior portion of the eye. It is a transitional zone and not a region specialised for high acuity central vision with densely packed cone cells. Option C is incorrect because the macula lutea is the broader yellowish area that provides the clearest overall vision, but the question specifically asks about the pinpoint centre containing only cones. The macula surrounds the fovea rather than being the exact point. The question describes the spot where vision is sharpest, a small depression at the centre of the macula containing only cones, no rods. That's the fovea centralis. It's a tiny pit where cone photoreceptors are packed the tightest, giving you maximum visual acuity. When you focus on reading these words, the image lands on your fovea. The optic disc is the blind spot, where ganglion cell axons bundle together and exit the eye as the optic nerve. It has 0 photoreceptors. The ora serrata is the jagged border where the retina ends anteriorly. It's a transition zone, not a vision centre. The macula lutea is the broader yellowish area around the fovea that provides clear central vision, but the question asks about the pinpoint centre with only cones. That's specifically the fovea. Here's a WGU D312 OA exam tip for you. Sharpest vision, centre of macula, only cones equals fovea centralis. If the question says maximum visual acuity or pinpoint depression, that's the fovea. Quick hook. Fovea equals sign focused vision at the centre. 34. A patient is diagnosed with elevated pressure inside the eye, and the physician explains that a clear, watery fluid produced in the eye is not draining properly through its normal exit channel at the junction of the cornea and sclera. Which fluid is accumulating and contributing to the increased intraocular pressure? A. Aqueous humour in the anterior cavity. B. Vitreous body in the posterior cavity. C. Cerebrospinal fluid from the meninges. D. Tears from the lacrimal gland. Can you guess the answer? Correct answer. A. Aqueous humour in the anterior cavity. Explanation. Option A is correct because aqueous humour is the clear, watery fluid produced by the ciliary body that fills the anterior cavity. It normally drains through the scleral venous sinus at the cornea-sclera junction and maintains intraocular pressure. Option B is incorrect because the vitreous body is a transparent jelly that fills the posterior cavity between the lens and retina. It is formed during embryonic development and is not renewed or drained through any exit channel during adult life. Option C is incorrect because cerebrospinal fluid is produced by the choroid plexus within the ventricles of the brain and surrounds the brain and spinal cord. It is not found within the eye and does not contribute to intraocular pressure. Option D is incorrect because tears are produced by the lacrimal gland and flow across the external eye surface to moisten and protect it. Tears drain through the nasolacrimal duct into the nasal cavity, not through internal eye drainage channels. The question describes the spot where vision is sharpest, a small depression at the centre of the macula containing only cones, no rods. That's the fovea centralis. It's a tiny pit where cone photoreceptors are packed the tightest, giving you maximum visual acuity. When you focus on reading these words, the image lands on your fovea. The optic disc is the blind spot, where ganglion cell axons bundle together and exit the eye as the optic nerve. It has 0 photoreceptors. The ora serrata is the jagged border where the retina ends anteriorly. It's a transition zone, not a vision centre. The macula lutea is the broader yellowish area around the fovea that provides clear central vision. But the question asks about the pinpoint centre with only cones. That's specifically the fovea. Here's a WGU D312 OA exam tip for you. Sharpest vision, centre of macula, only cones equals fovea centralis. If the question says maximum visual acuity or pinpoint depression, that's the fovea. Quick hook. Fovea equals sign focused vision at the centre. 35. A patient can move their eyes up, down and inward without any difficulty, but they are completely unable to move one eye outward to look toward the lateral side. The physician determines that the muscle responsible for pulling the eye laterally has lost its nerve supply. Which muscle has lost its innervation? A. The superior rectus muscle. B. The lateral rectus muscle. C. The medial rectus muscle. D. The inferior oblique muscle. Do you know the answer? Correct answer, B. The lateral rectus muscle. Explanation. Option B is correct because the lateral rectus is the only extrinsic eye muscle that abducts the eye, pulling it laterally to look outward. It is innervated by cranial nerve 6, and loss of this nerve supply prevents lateral eye movement. Option A is incorrect because the superior rectus muscle elevates the cornea, allowing the eye to look upward. Since the patient can look up without difficulty, this muscle and its nerve supply from cranial nerve 3 are functioning normally. Option C is incorrect because the medial rectus muscle adducts the eye, pulling it inward toward the nose. Since the patient can move the eye inward without difficulty, this muscle is not the one that has lost its innervation. Option D is incorrect because the inferior oblique muscle elevates the cornea and is innervated by cranial nerve 3. Its function is related to upward and rotational eye movement, not the lateral abduction that the patient is unable to perform. The patient can look up, down and inward but cannot look outward, can't abduct the eye laterally. The muscle responsible for pulling the eye to the side is the lateral rectus. It's the only muscle that abducts the eye. Since every other direction works, the other muscles and their nerves are fine. The superior rectus elevates the eye. Looking up works, so it's not the problem. The medial rectus adducts the eye inward. That works too. The inferior oblique elevates the eye with some rotation. Also working. The lateral rectus is innervated by CN6, the abducens nerve, and losing that nerve supply is why the eye can't look laterally. Here's a WGU D312 OA exam tip for you. This is the same concept as question 27, but asked from the muscle side instead of the nerve side. Can't abduct the eye equals lateral rectus problem. Quick hook. Lateral rectus equals sign looks laterally. Only one muscle does this job. 36. Sound waves enter the ear and travel through the ear canal until they strike a thin membrane that vibrates in response to the incoming pressure changes. These vibrations are then transmitted to the first of 3 tiny bones in the middle ear for further conduction. Which structure vibrates first when sound waves reach it? A. The oval window at the cochlea. B. The stapes bone in the middle ear. C. The tympanic membrane at the canal end. D. The round window releasing pressure. Do you know the answer? Correct answer, C. The tympanic membrane at the canal end. Explanation. Option C is correct because the tympanic membrane, also called the eardrum, is the thin structure at the end of the external auditory meatus that vibrates when sound waves strike it. It is the first membrane to respond to incoming sound. Option A is incorrect because the oval window receives vibrations from the stapes, the third bone in the middle ear, and transmits them to the inner ear. Option B is incorrect because the stapes is the last of the 3 bones in the middle ear, receiving vibrations after the malleus and incus. Option D is incorrect because the round window relieves pressure from vibrating fluid waves within the cochlea, responding after vibrations have passed through the middle ear. Sound waves travel down the ear canal and hit a thin membrane that vibrates. That's the tympanic membrane, the eardrum. It's the very first structure that vibrates when sound arrives. After the eardrum vibrates, it passes those vibrations to the malleus, which is the first ossicle physically attached to it. Then the malleus passes them to the incus, and the incus to the stapes. The stapes pushes against the oval window, transmitting vibrations into the inner ear fluid. The oval window comes at the end of the chain, not the beginning. The stapes is the third and last ossicle. It receives vibrations after the malleus and incus have already done their work. The round window is a pressure relief membrane in the cochlea that flexes after fluid vibrations pass through. It's the last stop, not the first. Here's a WGU D312 OA exam tip for you. The sound path goes eardrum first, then malleus, incus, stapes, oval window. The tympanic membrane is always the first thing to vibrate. Quick hook, think TM-MIS-O. Tympanic membrane, malleus, incus, stapes, oval window. 37. An audiologist explains to a patient that hearing depends on specialized receptor cells located within a specific organ sitting on the basilar membrane inside the spiral cochlear duct. These receptor cells convert sound vibrations into nerve impulses sent along the cochlear branch of cranial nerve 8. Which structure houses these receptor cells? A. The vestibule between ear chambers. B. The semicircular canals for balance. C. The auditory tube to the nasopharynx. D. The organ of Corti in the cochlea. Do you know the answer? Correct answer, D. The organ of Corti in the cochlea. Explanation. Option D is correct because the organ of Corti sits within the cochlear duct on the basilar membrane and contains hair cells that are the specialized auditory receptors. These cells convert sound vibrations into neural impulses transmitted via cranial nerve 8. Option A is incorrect because the vestibule is the area between the tympanic cavity and the cochlea that contains the saccule and utricle. These structures detect head position and linear movement for balance, not the sound vibrations needed for hearing. Option B is incorrect because the semicircular canals contain mechanoreceptors that detect rotational acceleration and deceleration of the head. They serve the vestibular balance system and do not house the hair cells responsible for hearing. Option C is incorrect because the auditory tube connects the middle ear to the nasopharynx for the purpose of equalizing air pressure across the tympanic membrane. It does not contain any sensory receptor cells involved in converting sound into neural signals. The question asks where the specialized hearing receptor cells sit. Hair cells on the basilar membrane inside the cochlea, sending signals along CN7. That's the organ of Corti. It's the actual hearing organ. Hair cells within it are the sensory receptors that convert sound vibrations into nerve impulses. The vestibule is the chamber between the middle ear and the cochlea containing the saccule and utricle. Those detect head position and linear movement for balance, not sound. The semicircular canals house the cristae ampullaris for detecting rotational head movement. Again, balance, not hearing. The auditory tube connects the middle ear to the nasopharynx to equalize pressure. It has no sensory cells at all. Here's a WGU D312 OA exam tip for you. Hearing receptor cells on the basilar membrane equals organ of Corti. If a question mentions hair cells and the cochlea together, that's your answer. Quick hook. Corti equal sign cochlea's hearing organ. Hair cells in Corti here. 38. A patient reports feeling dizzy when tilting their head forward or backward and has difficulty sensing whether they are standing upright relative to gravity. The physician suspects a problem with the sensory structures that detect linear acceleration and the static position of the head. Which receptor structures detect head position relative to gravity? A. Maculae within the vestibule organs. B. Cristae in the semicircular canals. C. Hair cells inside the organ of Corti. D. Ossicles within the middle ear space. Do you know the answer? Correct answer, A. Maculae within the vestibule organs. Explanation. Option A is correct because the maculae are sensory epithelium located within the saccule and utricle of the vestibule. They contain mechanoreceptors that detect the position of the head relative to gravity and linear acceleration or deceleration. Option B is incorrect because the cristae ampullaris are mechanoreceptor ridges located inside the ampullae of the semicircular canals. They detect rotational or angular acceleration of the head, which is dynamic equilibrium, not static position sensing. Option C is incorrect because the hair cells inside the organ of Corti are auditory receptor cells that respond to sound vibrations transmitted through the cochlear fluid. They serve hearing, not the detection of head position relative to gravity. Option D is incorrect because the ossicles are the three tiny bones in the middle ear that mechanically transmit sound vibrations from the tympanic membrane to the oval window. They have no sensory function for detecting body position or equilibrium. Dizzy when tilting the head, can't tell if they're standing upright. That's a problem with static equilibrium. Static equilibrium detects the position of the head relative to gravity and linear acceleration. The structures responsible are the maculae, sensory epithelium located inside the saccule and utricle in the vestibule. They contain mechanoreceptors that signal the brain about head position so you can maintain balance and posture. And the cristae ampullaris are in the semicircular canals and detect rotational movement. That's dynamic equilibrium, not static position sensing. Hair cells in the organ of Corti are for hearing, not balance. The ossicles transmit sound through the middle ear. They have no sensory function for detecting body position. Here's a WGU D312 OA exam tip for you. Static equilibrium and gravity detection equals maculae in the vestibule. Dynamic equilibrium and rotation equals cristae in the semicircular canals. Quick hook. Maculae equal sign gravity map. Cristae equal sign rotation circles. 39. A patient experiences severe vertigo during rapid head turning and rotation. The physician explains that mechanoreceptors within swellings near the base of 3 fluid-filled canals detect rotational head movement. Inside these swellings, a gelatinous mass sits atop hair cells and bends when the fluid shifts. Which structures contain these specific mechanoreceptors for rotational detection? A. The maculae inside the utricle organ. B. The organ of Corti on the membrane. C. The cristae ampullaris in the ampullae. D. The cochlear duct within the spiral. What do you think is the answer? Correct answer, C. The cristae ampullaris in the ampullae. Explanation. Option C is correct because the cristae ampullaris are saddle-shaped ridges of mechanoreceptors located within the ampullae of the 3 semicircular canals. The cupula sits atop the hair cells and bends when endolymph shifts during rotational head movement. Option A is incorrect because the maculae are sensory receptors inside the utricle and saccule that detect static head position and linear acceleration relative to gravity. They do not respond to the rotational acceleration described in this patient scenario. Option B is incorrect because the organ of Corti is the hearing receptor organ located on the basilar membrane inside the cochlear duct. It converts sound vibrations into neural impulses and has no function related to detecting head rotation or balance. Option D is incorrect because the cochlear duct is the membranous tube within the bony cochlea that contains the organ of Corti for hearing. It is part of the auditory pathway and does not contain the mechanoreceptors that sense rotational head movement. Vertigo during rapid head turning. The problem is with detecting rotational movement. That's dynamic equilibrium, handled by the cristae ampullaris inside the ampullae of the 3 semicircular canals. Each canal has a swelling, called an ampulla, near its opening into the utricle. Inside each ampulla sits a saddle-shaped ridge, the crista ampullaris, covered in hair cells. A gelatinous mass called the cupula sits on top of those hair cells. When the head rotates, endolymph lags behind slightly, dragging over the crista and bending the cupula and its embedded stereocilia, generating a receptor potential sent to the brain via CNC8. The maculae are in the utricle and saccule for gravity and linear motion, static equilibrium, not rotation. The organ of Corti handles hearing. The cochlear duct is part of the auditory system, not the balance system. Here's a WGU D312 OA exam tip for you. Rotational head movement plus semicircular canals equals cristae ampullaris. If the question mentions vertigo during turning or spinning, think cristae. Quick hook. Cristae and canals detect circular motion. 40. After sound waves pass through all 3 middle ear ossicles, the final bone in the chain pushes against a membrane-covered opening that transmits vibrations into the fluid-filled inner ear for processing by auditory receptors. Which ossicle is the last bone in the chain that contacts this inner ear opening? A. The malleus attached to the eardrums. B. The stapes contacting the oval window. C. The incus sitting between the others. D. The modulus at the cochlear centre. What do you think is the answer? Correct answer, B. The stapes contacting the oval window. Explanation. Option B is correct because the stapes, also called the stirrup, is the third and final bone in the ossicular chain. It contacts the oval window and pushes against it to transmit sound vibrations into the fluid of the inner ear cochlea. Option A is incorrect because the malleus is the first bone in the ossicular chain, attached directly to the tympanic membrane. It receives vibrations from the eardrum and passes them to the incus, not directly to the inner ear opening. Option C is incorrect because the incus is the second bone in the middle ear chain, sitting between the malleus and the stapes. It transmits vibrations onward to the stapes, but does not itself contact the oval window of the inner ear. Option D is incorrect because the modulus is the central bony pillar of the cochlea, around which the spiral turns of the bony labyrinth coil. It is a structural component of the inner ear, not one of the three ossicles in the middle ear chain. After sound passes through all three ossicles, the last bone in the chain pushes against a membrane-covered opening into the inner ear. That last bone is the stapes, the stirrup. It contacts the oval window, and when it pushes, vibrations transfer into the fluid-filled cochlea, where the organ of Corti converts them into nerve impulses. The malleus is the first ossicle, attached directly to the eardrum. It starts the chain, not the end. The incus sits in the middle between the malleus and stapes. It passes vibrations along but doesn't touch the oval window. The modulus is the central bony pillar of the cochlea that the spiral wraps around. It's a structural part of the inner ear, not an ossicle at all. Here's a WGU D312 OA exam tip for you. The ossicle chain runs malleus, incus, stapes. The stapes is always the last and it always contacts the oval window. Quick hook, stapes =, stirrup =, steps onto the oval window last. 41. A forensic examiner is studying a skull and notes that one specific cranial bone joins all the other cranial bones together and forms part of the skull base, orbital floor, and orbital walls. No other cranial bone makes contact with as many surrounding bones. Which cranial bone is the examiner identifying? A. The frontal bone forming the forehead. B. The occipital bone at the posterior skull. C. The parietal bone on the upper lateral skull. D. The sphenoid bone at the skull base. What do you think is the answer? Correct answer, D. The sphenoid bone at the skull base. Explanation: option D is correct because the sphenoid bone joins all the other cranial bones together and forms the base of the skull, as well as portions of the orbital floor and walls. No other single cranial bone articulates with so many neighbouring bones. Option A is incorrect because the frontal bone forms the forehead and the anterior cranial fossa, but does not join all other cranial bones. It articulates with the parietal bones at the coronal suture, but does not contact every cranial bone. Option B is incorrect because the occipital bone forms the posterior skull and the posterior base of the cranial cavity. While it articulates with several bones, it does not join all cranial bones together or form parts of the orbital walls. Option C is incorrect because the parietal bones form most of the upper lateral skull and articulate with the frontal, temporal, occipital, and sphenoid bones. However, they do not join all cranial bones or contribute to the orbital floor structure. This question asks you to identify the one cranial bone that touches every other cranial bone. That's the sphenoid. It sits at the base of the skull and connects to the frontal, parietal, temporal, occipital, and ethmoid bones. No other cranial bone has that many articulations. On top of that, it forms parts of the orbital floor and orbital walls, the bony socket around the eye. The frontal bone makes the forehead and articulates with the parietals at the coronal suture, but it doesn't reach every other cranial bone. The occipital bone forms the back of the skull and the posterior base, but it misses contact with several anterior bones. The parietal bones cover the upper sides of the skull and meet the frontal, temporal, occipital, and sphenoid, but they don't reach the ethmoid or form orbital structures. Here's a WGU D312 OA exam tip for you. If a question says joins all cranial bones or articulates with the most bones, that's the sphenoid. Quick hook. Sphenoid =, spider bone. It sits in the middle and reaches out to touch everything. 42. A radiologist reviewing a spinal image identifies the 5 largest vertebrae in the lower back region that bear the greatest amount of body weight compared to all other vertebral segments. These vertebrae sit between the thoracic region above and the sacrum below. Which vertebral region contains these largest weight-bearing vertebrae? A. The lumbar region with 5 vertebrae. B. The cervical region with 7 vertebrae. C. The thoracic region with 12 vertebrae. D. The sacral region fused into one bone. What do you think is the answer? Correct answer, A, the lumbar region with 5 vertebrae. Explanation. Option A is correct because the lumbar vertebrae are the 5 largest vertebrae in the spinal column, located in the lower back. They bear the most body weight of all vertebral segments due to their position supporting the upper body. Option B is incorrect because the cervical region contains 7 smaller vertebrae in the neck that support the head. They bear far less weight than the lumbar vertebrae and are the smallest vertebrae in the entire spinal column. Option C is incorrect because the thoracic region contains 12 vertebrae that articulate with the ribs. While they are larger than cervical vertebrae, they do not bear as much weight or have the massive size of the lumbar vertebrae. Option D is incorrect because the sacrum is a single bone fused from 5 embryonic vertebrae at the base of the spine. While it transfers weight to the pelvis, the question specifically describes unfused vertebrae in the lower back region. 5 vertebrae in the lower back, the largest in the entire spinal column, bearing the most body weight. That's the lumbar region, L1 through L5. They sit between the thoracic vertebrae above and the sacrum below, and because everything above them pushes down, they're built big to handle the load. The cervical region has 7 vertebrae in the neck, C1 through C7. They're the smallest, supporting just the head. The thoracic region has 12 vertebrae, T1 through T12, that articulate with the ribs. They're larger than cervical, but still not as massive as lumbar. The sacrum is fused from 5 embryonic vertebrae into one bone. It transfers weight to the pelvis, but the question specifically says unfused vertebrae in the lower back, which rules out the sacrum. Here's a WGU D312 OA exam tip for you. Largest vertebrae plus lower back plus most weight-bearing equals lumbar. Remember the vertebral formula? 7 cervical, 12 thoracic, 5 lumbar, 1 sacrum, 1 coccyx. Quick hook, lumbar equals sign lumbar, big heavy logs in the lower back. 43. A medical student studying a skull specimen traces a line of articulation running along the midline at the top of the skull from front to back. This suture unites the left and right parietal bones at the very top of the cranium along the superior midline. Which cranial suture follows this specific path? A, the coronal suture at the front. B, the lambdoid suture at the back. C, the sagittal suture along the midline. D, the squamous suture on the side. Do you know the answer? Correct answer, C, the sagittal suture along the midline. Explanation. Option C is correct because the sagittal suture runs along the midline at the top of the skull, uniting the 2 parietal bones. It extends from the coronal suture anteriorly to the lambdoid suture posteriorly along the superior cranial surface. Option A is incorrect because the coronal suture runs side to side across the skull, separating the frontal bone from the 2 parietal bones. It does not follow the midline from front to back at the top of the cranium. Option B is incorrect because the lambdoid suture extends downward and laterally from the junction with the sagittal suture, joining the occipital bone to the parietal and temporal bones at the posterior skull, not along the midline top. Option D is incorrect because the squamous suture sits on the lateral skull, uniting the temporal bone with the parietal bone on each side. It does not run along the midline and is positioned much lower than the top of the cranium. A suture running along the midline at the top of the skull from front to back, uniting the left and right parietal bones. That's the sagittal suture. It runs in a straight line from the coronal suture at the front to the lambdoid suture at the back, right along the superior midline of the cranium. The coronal suture goes side to side across the skull, separating the frontal bone from the parietal bones. It doesn't follow the midline front to back. The lambdoid suture angles downward and laterally at the posterior skull, joining the occipital bone to the parietals and temporals. It's at the back, not along the top midline. The squamous suture sits low on the lateral skull where the temporal bone meets the parietal bone on each side, nowhere near the midline or the top. Here's a WGU D312 OA exam tip for you. Midline, top of skull, unites two parietal bones equals sagittal suture. Think of the word sagittal, it means arrow. An arrow shot from front to back along the top of the skull follows the sagittal suture. Quick hook. Sagittal equals sine arrow straight down the middle. 44. A trauma surgeon evaluating a patient with chest injuries explains that the sternum consists of 3 distinct parts stacked vertically from top to bottom, and that this bone, along with 12 pairs of ribs, forms a protective cage. Which answer correctly lists the 3 parts of the sternum from superior to inferior? A. Xiphoid process body and manubrium. B. Body, manubrium and xiphoid process. C. Manubrium, xiphoid process and body. D. Manubrium, body and xiphoid process. What do you think is the answer? Correct answer. D. Manubrium, body and xiphoid process. Explanation. Option D is correct because the sternum from superior to inferior consists of the manubrium at the top, the body in the middle, and the xiphoid process at the bottom. Together with 12 pairs of ribs, these form the thoracic cage protecting vital organs. Option A is incorrect because it reverses the correct order, listing the xiphoid process first at the top. The xiphoid process is the smallest inferior portion of the sternum, not the superior part, and the manubrium sits at the top. Option B is incorrect because it places the body first and the manubrium second, which reverses the top 2 components. The manubrium is the superior portion that articulates with the clavicles, while the body is the middle segment below it. Option C is incorrect because it places the xiphoid process between the manubrium and body, which is not the correct anatomical order. The body sits between the manubrium and xiphoid process, occupying the largest middle portion of the sternum. The sternum has 3 parts stacked top to bottom: manubrium, body, xiphoid process. The manubrium is the superior piece. It's the widest part at the top, and it articulates with the clavicles. The body is the long middle segment. The xiphoid process is the small pointed projection at the very bottom. Option A flips the order entirely, putting the xiphoid at the top. Option B puts the body first and the manubrium second. Wrong sequence for the top 2 pieces. Option C sandwiches the xiphoid between the manubrium and the body, which doesn't match anatomy. The correct order from top to bottom is always manubrium, body, xiphoid process. Here's a WGU D312 OA exam tip for you. The sternum order from superior to inferior is manubrium, body, xiphoid. If you see a question asking about sternum parts, check the sequence carefully. The distractors just rearrange the same 3 words. Quick hook. MBX from top to bottom. My body's extra protected. 45. During a facial examination, a physician palpates the prominent cheekbone structure on each side of the patient's face and explains that this paired bone forms the cheek prominence and contributes to the lateral orbital wall. The bony arch extending from this region toward the ear is also named after it. Which facial bone is the physician describing? A. The zygomatic bone forming the cheek. B. The maxilla forming the upper jaw. C. The nasal bone forming the nose bridge. D. The lacrimal bone near the eye corner. Do you know the answer? Correct answer, A. The zygomatic bone forming the cheek. Explanation. Option A is correct because the zygomatic bones form the cheek prominences and contribute to the lateral orbital walls. The zygomatic arch, a bridge of bone extending from the cheek toward the ear canal, is named after this paired facial bone. Option B is incorrect because the maxilla forms the upper jaw and contributes to the floor of the orbits and the roof of the mouth. It is not the bone responsible for the cheek prominence or the arch extending toward the ear. Option C is incorrect because the nasal bones are small paired bones that form the bridge of the nose at the midline of the face. They do not form the cheek prominences or contribute to the lateral bony arch near the ear. Option D is incorrect because the lacrimal bones are tiny paired bones sitting at the medial corner of each eye orbit near the tear ducts. They are far too small and medially positioned to form the prominent cheekbone structure palpated on the lateral face. The physician palpates the cheekbone and describes a paired bone forming the cheek prominence, contributing to the lateral orbital wall with a bony arch extending toward the ear named after it. That's the zygomatic bone. The zygomatic arch stretches from the cheek toward the ear canal. You can feel it on yourself right now if you press along that ridge. The maxilla forms the upper jaw and contributes to the orbital floor and the roof of the mouth. But it's not the cheekbone and doesn't have an arch named after it running toward the ear. The nasal bones are small and form the bridge of the nose, nowhere near the cheek prominence. The lacrimal bones are tiny, sitting at the medial corner of each orbit near the tear ducts, way too small and too medially placed to be the prominent cheekbone. Here's a WGU D312 OA exam tip for you. Cheekbone plus zygomatic arch equals zygomatic bone. The name zygomatic shows up in both the bone and the arch. That's your clue. Quick hook. Zygomatic =sign cheekbone zone. 46. A student examining a tongue model notices that the largest papillae are arranged in a V-shaped row at the back of the tongue, and that the nerve carrying taste information from this region travels to the brainstem. Which type of papillae are arranged in this distinctive V-shaped pattern at the posterior tongue? A. Fungiform papillae on the surface. B. Velator papillae at the tongue back. C. Filiform papillae across the tongue. D. Foliate papillae on the lateral edge. What do you think is the answer? Correct answer, B. Velat papillae at the tongue back. Explanation. Option B is correct because valate papillae are the largest type of lingual papillae and are arranged in a characteristic V shape at the back of the tongue. They contain taste buds and are innervated by the glossopharyngeal nerve for taste sensation. Option A is incorrect because fungiform papillae are knob-like projections scattered across the tongue surface. They are smaller than valate papillae and are distributed more broadly rather than forming a V-shaped row at the posterior tongue. Option C is incorrect because filiform papillae are thread-like elevations that cover most of the tongue surface and provide texture for food manipulation. Importantly, filiform papillae do not contain any taste buds at all and cannot detect flavours. Option D is incorrect because foliate papillae are located on the lateral posterior edges of the tongue, not arranged in a V shape at the back. They contain taste buds but are positioned along the sides rather than in the distinctive posterior row. Largest papillae, arranged in a V shape at the back of the tongue. Those are valate papillae. There are only about 8 to 12 of them, but they're the biggest and they form that distinctive V-shaped row separating the anterior 2/3 of the tongue from the posterior third. They contain taste buds innervated by the glossopharyngeal nerve, CN9. Fungiform papillae are the knob-shaped bumps scattered across the tongue surface. They have taste buds, but they're smaller and don't form a V-shape. Filiform papillae are the thread-like projections covering most of the tongue that give it texture and help grip food. Here's the critical detail. Filiform papillae contain no taste buds at all. They can't detect flavour. Foliate papillae sit along the lateral posterior edges of the tongue, not in a V shape at the back. Here's a WGU D312 OA exam tip for you. V shape at the back of the tongue equals valate. And remember that filiform is the odd one out. No taste buds. Quick hook. Valate =sign V shape. Filiform =sign feels but doesn't flavour. 47. After taste receptor cells in the tongue detect dissolved food molecules, the gustatory signals travel through cranial nerves to the brainstem and then must pass through a specific relay station in the diencephalon before reaching the primary gustatory cortex in the parietal lobe. Which brain structure serves as this relay station for taste information? A. The hypothalamus below the thalamus. B. The limbic system for emotional memory. C. The thalamus in the diencephalon. D. The cerebellum at the posterior brain. Do you know the answer? Correct answer, C. The thalamus in the diencephalon. Explanation? Option C is correct because taste information travels from the gustatory nucleus in the brainstem to the thalamus, which serves as the primary relay station in the diencephalon. The thalamus then projects this gustatory information to the primary gustatory cortex in the parietal lobe. Option A is incorrect because the hypothalamus is located inferior to the thalamus and connects taste to emotional states and hunger signaling. While involved in the broader taste pathway, it is not the main relay station that sends taste signals to the gustatory cortex. Option B is incorrect because the limbic system is involved in connecting taste experiences to emotional states and memory formation. It processes the emotional aspects of taste, but is not the relay station that routes taste signals to the primary gustatory cortex. Option D is incorrect because the cerebellum is located at the posterior base of the brain and is primarily involved in coordinating voluntary movement and maintaining balance. It does not function as a relay station in the gustatory sensory pathway. Taste signals travel from receptor cells through cranial nerves to the gustatory nucleus in the brainstem, and then they need a relay station before reaching the gustatory cortex in the parietal lobe. That relay station is the thalamus. The thalamus is the brain's grand switchboard. Almost all sensory information passes through it on the way to the cortex. Taste is no exception. The hypothalamus sits below the thalamus and connects taste to hunger and emotional states, but it's not the relay that routes signals to the gustatory cortex. The limbic system ties taste to emotions and memories, important for how you feel about food, but not the sensory relay. The cerebellum coordinates movement and balance at the posterior brain. It plays no part in the taste pathway. Here's a WGU D312 OA exam tip for you. For nearly every sensory pathway, the thalamus is the relay station to the cortex. Taste, vision, hearing, touch, they all pass through the thalamus. The one big exception is smell, which goes to the cortex more directly. Quick hook. Thalamus equals sign. The relay for almost everything sensory. 48. Olfactory receptor cells in the nasal cavity detect airborne odour molecules and generate neural impulses. Their axons must pass through a sieve-like bony structure separating the nasal cavity from the anterior cranial fossa before reaching the olfactory bulbs above. Which structure do these nerve fibres pass through? A. The cribriform plate of the ethmoid. B. The foramen magnum of the occipital. C. The nasal septum dividing the cavity. D. The hard palate below the nasal cavity. What do you think is the answer? Correct answer, A. The cribriform plate of the ethmoid. Explanation. Option A is correct because the cribriform plate is a sieve-like portion of the ethmoid bone that separates the nasal cavity from the anterior cranial fossa. Olfactory nerve fibres pass through its tiny perforations to reach the olfactory bulbs above. Option B is incorrect because the foramen magnum is the large opening in the occipital bone at the base of the skull where the spinal cord enters the cranium. It is located at the posterior skull base, not between the nasal cavity and the anterior fossa. Option C is incorrect because the nasal septum is a vertical partition that divides the nasal cavity into left and right halves. It is formed by the vomer and ethmoid perpendicular plate, but does not allow nerve fibres to pass through. Option D is incorrect because the hard palate is the bony floor of the nasal cavity that separates it from the oral cavity below. Olfactory nerve fibres travel upward through the roof of the nasal cavity, not downward through the palate. Olfactory receptor cell axons need to get from the nasal cavity up to the olfactory bulbs in the anterior cranial fossa. The bony barrier between these two spaces is the cribriform plate of the ethmoid bone. It's perforated with tiny holes, sieve-like, and the nerve fibres thread through those perforations to reach the bulbs above. The foramen magnum is the large opening in the occipital bone at the posterior skull base, where the spinal cord passes through. It's at the back and bottom, nowhere near the nasal cavity. The nasal septum divides the nasal cavity into left and right halves, using the vomer and the perpendicular plate of the ethmoid. It's a vertical wall, not a horizontal sieve with holes for nerves. The hard palate is the bony floor of the nasal cavity, separating it from the mouth below. Olfactory fibres travel upward through the roof, not downward through the floor. Here's a WGU D312 OA exam tip for you. Olfactory nerve fibres passing from nose to brain equals cribriform plate of the ethmoid. The word cribriform literally means sieve-shaped. Quick hook. Cribriform equals sign sieve for smell nerve fibres. Cribriform equals sign form of a sieve. 49. A person enters a room with a strong perfume scent and notices the smell immediately, but after several minutes they can no longer detect the same odour, even though the perfume concentration has not changed. The olfactory epithelium has decreased its sensitivity to the sustained stimulus. Which term describes this gradual decrease in odour sensitivity? A. Olfactory transduction of the signal. B. Depolarization of the receptor cell. C. Receptor potential in the epithelium. D. Adaptation of the olfactory epithelium. Do you know the answer? Correct answer, D. Adaptation of the olfactory epithelium. Explanation. Option D is correct because adaptation refers to the olfactory epithelium's decreasing sensitivity to sustained odours over time. The receptor cells gradually stop responding as strongly when the same odorant is continuously present at constant concentration. Option A is incorrect because olfactory transduction is the initial process of converting chemical odour molecules in the air into neural impulses through local depolarization. It is the detection mechanism, not the gradual decrease in sensitivity over time. Option B is incorrect because depolarization of the receptor cell is the electrical change that occurs when an odorant molecule first stimulates the olfactory receptor. It is a single event in signal generation, not the progressive reduction in sensitivity. Option C is incorrect because a receptor potential is the graded electrical signal produced in a sensory receptor cell upon stimulation. It describes the initial electrical response to an odorant, not the declining sensitivity that develops after prolonged exposure. You walk into a room, smell perfume immediately, and 5 minutes later you don't notice it anymore, even though the concentration hasn't changed. That's olfactory adaptation. The receptor cells in the olfactory epithelium gradually decrease their response to a constant, sustained odour. Your nose basically says, "I got it, moving on," and stops sending the signal as strongly. Olfactory transduction is the initial conversion of a chemical odour molecule into a nerve impulse through depolarization. That's the detection event at the start, not the fading over time. Depolarization of the receptor cell is the single electrical event when an odorant first triggers the receptor. One moment in time, not a progressive decline. A receptor potential is the graded electrical signal produced when a receptor cell gets stimulated. Again, that's the initial response, not the gradual loss of sensitivity. Here's a WGU D312 OA exam tip for you. Smell something, then stop smelling it, even though it's still there, equals adaptation. If a question describes decreasing sensitivity over time, adaptation is the word they want. Quick hook. Adaptation equals sign your nose adjusts and stops noticing. 50. A histologist examining a cross-section of a tongue papilla identifies clusters of cells accessed through a tiny opening in the surface epithelium. Inside each cluster, 3 distinct cell types are visible including the main sensory cells with hair-like extensions projecting through the opening toward dissolved food molecules. Which cell type within the taste bud serves as the primary sensory detector? A. Basal cells regenerating other cell types. B. Supporting cells providing structure only. C. Gustatory receptor cells with gustatory hairs. D. Epithelial cells lining the tongue surface. What do you think is the answer? Correct answer, C. Gustatory receptor cells with gustatory hairs. Explanation. Option C is correct because gustatory receptor cells are the main sensory cells within taste buds. They have gustatory hairs that extend through the taste pore to contact dissolved food molecules, detecting the 5 basic taste sensations in the oral environment. Option A is incorrect because basal cells are small round stem cells located at the base of the taste bud. Their function is to regenerate the gustatory receptor cells and supporting cells, not to directly detect dissolved food molecules themselves. Option B is incorrect because supporting cells are long spindle-shaped cells within the taste bud that provide structural framework and mechanical support. They do not have sensory hairs extending through the taste pore for detecting taste molecules. Option D is incorrect because epithelial cells line the general tongue surface and form the outer covering of the papillae. They are not located inside the taste bud clusters and do not function as chemoreceptors for detecting dissolved food substances. A histologist sees clusters of cells inside a tongue papilla accessed through a tiny opening in the surface epithelium. That opening is the taste pore. Inside, 3 cell types are visible. The main sensory cells with hair-like extensions poking through the pore toward dissolved food molecules. Those are gustatory receptor cells with gustatory hairs. They're the primary taste detectors, sensing sweet, salty, sour, bitter, and umami by contacting dissolved taste-stunts in saliva. Basal cells sit at the base of the taste bud. They're small round stem cells that regenerate the other cell types when they wear out. They don't detect taste themselves. Supporting cells are long spindle-shaped cells that provide the structural framework inside the taste bud. They hold things together but have no sensory hairs and can't detect flavours. Epithelial cells line the general tongue surface outside the taste bud. They're part of the tongue's outer covering and don't function as chemoreceptors. Here's a WGU D312 OA exam tip for you. Primary sensory cell in the taste bud with hairs through the taste pore equals gustatory receptor cell. Know all 3 cell types. Gustatory receptor cells detect, supporting cells support, basal cells regenerate. Quick hook. Gustatory cells grab the taste through their hairs. You have reached the end of our first question set. For the free complete exam preparation, visit our website, link in the description, where you find thousands more practice questions and comprehensive study guides. Thanks for studying with us today. See you in the next video.